and Microgravity team. Their mentor was Becky Fritchie, and please join me in welcoming their team.
Hi, I'm Nathan Scaff, and I'm from Lexington, Kentucky.
Hello, I'm Jeremy Liu, and I'm from Carlsbad, California.
Hi, I'm Maya Gilhoff. I'm from the Cincinnati area in northern Kentucky.
Hi, I'm Johanna Donegan-Wers, and I'm from Chittenango, New York.
Hi, I'm Bryn Edwards, and I'm from Fort Lauderdale, Florida.
Hi, my name is Natalie Long, and I'm from Richmond, Virginia.
Hello, my name is Nathan Heath, and I'm from Malvern, Arkansas.
Hello, my name is Joyce Taylor, and I'm from Charlotte, North Carolina. Our mentorship consists of Zoe Zlatic, Becky Fritchie, Laura Tomlin, Dr. George Pantelos, and Ms. Phyllis Friola. They help by giving us advice and guidance throughout this entire project.
After designing the mission patch, I'll explain its symbolism. The octagon displays 8 sides to demonstrate the 8 members. Earth demonstrates how this topic will relate back to life on Earth and hopefully improve people's lives. The blood cells in orbit demonstrate how the research focuses on the characteristics of RBCs in microgravity. The NASA worm font, displayed on all the text, represents our ties back to NASA. The star pattern on the background is from the NASA meatball logo and also shows our connection to this organization. Lastly, the light blue ring also demonstrates our connection to Earth as it displays the colors of our ocean while complementing the patch and making it look more balanced.
This investigation will examine current anemia treatments, analyze their applicability for space hemolysis, and discuss how these treatments can be adapted to better suit spaceflight. Even though this is a new topic of research, there's a multitude of reasons for the cause of space hemolysis, including fluid shifts and more. We started looking into anemia treatments to treat space hemolysis because anemia and space hemolysis are so close, the treatments could potentially be interchangeable. So within our research, we will look into ways that anemia is cured and see if it's applicable to space hemolysis. The goal of this research is to find out how to further prevent health complications with blood throughout long space journeys. Our goal is to be able to see if anemia treatments are applicable to space hemolysis induced in microgravity. When exposed to a period in microgravity, it has been found that astronauts will experience a phenomenon called space hemolysis. During this process of hemolysis, astronauts will experience up to 45% of an increase in the destruction and the rupture of red blood cells or RBCs. The effect of hemolysis continues even after astronauts come back to the Earth setting. We're currently trying to solve this issue because any sort of anemia or anemia-like abnormality will not allow our body to get the sufficient amount of oxygen we need. It can start to damage our vital organs that are reliant on blood, like the heart and brain. Also, the body's tissues will also be starved of oxygen as well. This leads to hypoxia, which shuts down the necessary bodily functions.
So what is hemolysis? Hemolysis is when the destruction of red blood cells, or RBCs, exceeds the rate in which those RBCs are being replenished by bone marrow. Bone marrow being a spongy tissue which is found inside certain bones that produces red blood cells, among other things. Now, how do the bodies of astronauts respond to microgravity? Microgravity being one of the key factors as to why astronauts obtain this adaptation. In short, microgravity is the state in which objects appear to be in a constant state of free fall, because they are orbiting around Earth in a continuous rate. Bodies react to microgravity with an abnormal increase in hemoglobin degradation. Hemoglobin is a protein found in RBCs that carries oxygen from the lungs to the rest of the body and returns carbon dioxide to the lungs, or to the tissues back to the lungs. This is an essential function for transporting oxygen to the cells and allowing for cell respiration. Then there's hematocrit, which is the volume of blood that is occupied by healthy RBCs. When there's hematocrit degradation, it means the blood's oxygen carrying capacity has been reduced. Reducing plasma volume refers to the liquid component of the blood that contributes to 55% of the blood's total volume. When this is reduced, it jeopardizes the circulatory system. Lastly, reducing erythrocyte mass indicates a decrease in the total number of red blood cells. Overall, these bodily responses to microgravity impair the oxygen carrying capacity of the blood. Moving into how space anemia affects the body. For one, there is limited oxygen transportation. This is the main source for the other symptoms, those being fatigue or lack of energy, dizziness, potentially headaches, decreased strength and feeling weaker, abnormal heart functions, which can be heart palpitations, disrupted cognitive ability, so the processes of thinking, observing, and solving are more difficult, shortness of breath, and many more. So what do all these physiological responses lead to? Astronauts' anemia. This is also synonymously referred to as space anemia and microgravity-induced anemia. When hemolysis exceeds a rate of 54%, we know there's a significant imbalance. Reasons for astronauts experiencing this adaptation include factors like radiation exposure, fluid distribution, and microgravity, which our main focus for the project will be on microgravity. Radiation exposure is emission of energy as electromagnetic waves, and it damages RBCs. On the other hand, fluid distribution is the movement and redistribution of bodily fluids due to the absence of gravity, which alters blood volume. Another factor is extended missions with prolonged periods of time in microgravity, which continues to add to this problem's severity.
Measuring and monitoring space hemolysis. In order to properly examine the effects of anemia treatments in astronauts, there needs to be a way to test for anemia. Right now, the most common way to test for anemia is using blood draws. However, for spaceflight, there's an increased risk of complications with this technique, and it requires a large amount of materials to be carried on missions, such as sterilized needles, needle disposal, syringe, blood storage, and testing equipment. An alternative method to this is using a deep learning AI. A study was conducted with this where it analyzed retinal fundus images from the UK Biobank, composed over 114,205 fundus images. It found that this deep learning model was able to detect anemia and quantify blood hemoglobin levels with a 95% confidence rate. Fundus photography is something that's already aboard many space missions, including the ISS, and it can be done a lot more frequently than blood draws.
Possible causes for hemolysis. Microgravity is the most likely option given for these rates of increase are unique to spaceflight. Next, fluid shift is possible due to the body purging off 10% of its fluid volume while under the effects of microgravity. Next, other theories being discussed include radiation, isolation, and confinement. Space hemolysis is a catastrophe. Space hemolysis proves itself to be a major difficulty for astronauts due to its similar effects on the body to anemia. While currently unsolved, looking to treatments for anemia could aid in helping cure space anemia. Space hemolysis does not only affect astronauts while under the influence of microgravity, but effects can extend for around a year after astronauts return to Earth, depending on how long they spent under the effects of microgravity. Severe effects include heart failure, loss in control of red blood cell count, which can lead to unwanted structural changes in organ structure, and a loss of consciousness.
Current solutions have been discovered by space organizations to combat hemolysis, which is when the body destroys red blood cells for the duration someone is in space. For example, astronauts who exercise the upper body will face changes in their fluid dynamics. Not only do astronauts need an improved fitness plan to compensate for microgravity, they will need to enhance their nutrition and vitamin intake. It will also help to understand how the bloodstream works in microgravity. Additionally, pharmaceutical treatments are being tested to see if they can undergo the conditions in space to accommodate the astronauts' health for their neurological and blood systems. Although these results for these treatments aren't guaranteed, scientists at NASA and beyond have numerous studies supporting this research.
Alright, so this image below is one of the two ZQubes developed by Bob Twiggs. This version is a 10 by 10 by 20 centimeter rectangle, equipped with a 3D printed exterior shell, a data logger module, a mini testing container, and a mini camera. The cube also has multiple other sensors that they can measure, as it's equipped with gyroscopes and accelerometers, and much more. Another interesting additional fact is that the ZQube was actually inspired by a Beanie Baby box, which Mr. Twiggs found when shopping. In our ZQube, we have 3 tubes of liquid: blood, water, and urine. The blood and urine are solutions. The blood we made with a mixture of glycerin and water, and the urine out of salt and water. We first reached out to Dr. Pantelos for a blood solution. Once we researched the rest of the liquids, we syringed the solutions into separately labeled tubes. On our final prototype, the different solutions were dyed to ensure that we can identify each of them differently in space. Our main goal is to use the camera feature of the ZQube to measure the visual changes in the liquids. Specifically, we want to notice the viscosity, which is the fluid's resistance to flow. This information is critical to know how these human bodily fluids may perform in microgravity. For example, it will identify how these liquids perform when attempting something such as a surgical procedure. Now, we expect all the liquids to stick to the walls of our testing tubes. This is due to the adhesion of the liquids. Considering the viscosity, we also believe that blood will adhere more to the container than the water. Urine and water are expected to perform around the same, as not only is our solution mainly water, but so is the actual product.
Erythropoietin-stimulating agents, or ESAs, are a glycoprotein hormone that stimulates red blood cell production. ESA is a pharmacological reproduction of EPO, created using recombinant DNA and manufactured using transfected Chinese hamster ovary cells, or CHOs. Currently, ESAs are approved for conditions in which RBC production is impaired. However, after some analysis, we concluded that ESA is likely to be an ineffective treatment for space hemolysis. This is because in space hemolysis, the immature red blood cells, or reticulocytes, as well as the mature ones, lack EPO receptors and cease to respond to EPO completely. This study defines space hemolysis as EPO independent. Therefore, a hormone replicating EPO is likely to be unable to stimulate red blood cell production.
So refrigerated blood in the medical world stored for blood transfusions can only be used for 42 days before basically expiring. The minute blood leaves the body, nitric oxide levels drop and continue to drop until the donation is over. After those 42 days, the gas is nearly non-existent and would not be able to deliver oxygen properly. When looking at this huge task of bringing blood to space for the off chance a blood transfusion is necessary, or even just revolutionizing storing blood on Earth, dehydrated blood offers the perfect solution. Dehydrated blood is exactly as it sounds, blood that has a liquid component has its liquid component removed and transforms into a dry pink powder that can potentially be rehydrated. To rehydrate this blood, sterile water must be added and mixed around. In space, there's a specific bag that would be necessary from the presence of microgravity because there's no hydrostatic pressure. This dual blood bag has the powder on one side and the sterile water on the other, so when it's time to combine the two, the notch in the middle is removed. Then to actually activate the solution, a pump is necessary as well as some manual mixing from outside of the bag. This process has been proved to be successful in small quantities, so the next challenge is moving forward and seeing if larger volumes can be taken be sufficient for the extent of the space travel. Rehydrated blood is thought to mend oxidative damage from microgravity by replenishing the nitric oxide levels and ensuring proper oxygen delivery to tissue. This solution addresses the challenge of storing and using blood in space, potentially providing a reliable way to manage anemia during extended missions.
The preliminary schedule consisted of first coming up with ideas regarding the experiment, and then receiving the ZQube and creating ideas to apply the subject. And after creating these ideas and applying them, we altered the project to see if there were any flaws. And lastly, we did more research and created the slides.
Possible causes of hemolysis in spaceflight include radiation, fluid shifts, and oxygen needs lowered. Treatments for this condition include surgery, blood transfusion, and supplements. In conclusion, we can see that hemolysis is one of the many problems we face as we venture more into space. This debilitating condition breaks down the red blood cells of astronauts at an increased rate of 54% in space compared to here on Earth. This causes anemia, which starves the vital organs of oxygen, leading to death if gone untreated. This is why it is critical for us to find a suitable treatment methods to use on spaceflights to assist the astronauts with this condition on long space journeys.
Our team has worked tirelessly on figuring out how hemolysis would react in microgravity. Our goal for this mission is to examine anemia treatments by studying hemolysis in microgravity. Our mission aligns with NASA's strategic goal of expanding human knowledge, capability, and opportunity in space. Future research could lead to discovering a treatment to alleviate hemolysis. Furthermore, learning about how fluid dynamics behave in the upper body will support the data behind why astronauts have swollen faces in space.
So we invite all members of the team to please turn on their cameras and their microphones. We have several questions from our Slido app. First of all, why did your team decide to focus on hemolysis?
I can answer that question. Our group decided on hemolysis due to the research on its effects and microgravity being relevant and it being a relatively new topic. Another reason for us choosing hemolysis is that the effects of space hemolysis are becoming more severe over longer duration missions and therefore needing a solution for longer journeys such as those to Mars made it more relevant.
Great. Would the blood transfusions through dehydrated blood require a specialist or a doctor to complete? And if so, would this be a requirement for all future missions?
I'll take that one. So there are a lot of people still working on the sort of concept of dehydrated blood. So I think a specialist would definitely be helpful on those missions, but I'm sure once it becomes more of a solidified solution, I think more people will be informed and know a lot about the topic. So it won't be a specific necessity for some missions.
Your next question is, what is anemia?
So I'll take that one too. There's a difference between anemia and space anemia. Anemia is the actual issue of not having enough healthy RBCs or red blood cells. And that includes the healthy hemoglobin protein that brings oxygen throughout the body. Space anemia, on the other hand, is induced by the astronauts spending prolonged periods of time in space or microgravity. The microgravity environment sort of is cause for those fluid shifts and then later less healthy red blood cells.
And what are the complications that can occur with blood draws in microgravity?
So one complication during blood draws is the actual bag and process that is used. In microgravity, there is, instead of blood is pooling, it sort of is released into the air to flow in unpredictable ways. So when giving blood, you'll need a pump rather than just be able to let the blood flow. And then I think, Jeremy, you can add on to that.
Well, yeah, like just to clarify, because of the adhesion and cohesion effects of liquid in microgravity, the blood will probably, and this is also what we test on the ZQube too, but the blood will kind of drip up this or like encapsulate the sides of the bag and encapsulate the sides of the tube, which can cause like slosh or bubbles or inconsistent flow of blood, which is why, as Britton stated, IV drips designed for use in space usually use mechanisms to push on the liquid rather than to rely on gravity.
What were some of the challenges your team had to overcome during the time at SEAS?
I'll go ahead and take this one.
But when we were working on this project, we faced a couple of issues working on topic because everybody wanted to be different. But with that, we ended up figuring the topic that interested us all, and then we broke up on different parts of research based on whatever our mission was.
And how has your experience at SEAS affected your moving into your junior year in high school?
I'll go ahead and take this one. I will say that going into the junior year with this internship has affected me in a positive way, such as it's rekindled my interest in the STEM field, as I've been really able to interact with other people with similar interests to me, and also with the materials needed to further that interest in the STEM field, such as studying the biology of human health, how it's affected in microgravity. It's been a really interesting topic, and I'm happy that I was able to really go into it more deeply during the internship.
Well, you got lots of thumbs up and great job, so congratulations to your team. Our next team is the In Situ resource team. Their project mentor is Stephanie Hanover, a middle and high school science teacher in Bryan, Texas. She is in her 15th year in science education. Stephanie is a passionate, lifelong learner who believes that students learn best through discovery and real-world experiences. And she works to bring those to the classroom and extracurricular settings every day. Please join me in welcoming the In Situ resource team.
Hello everyone, thank you for joining us today. We are the In Situ Resource Utilization Team. Our project, abbreviated as LEMUR, stands for Lunar Exploration Mission Using Regolith. Let's introduce the team.
Hello, my name is Jatin and I'm from Texas.
Hi, my name is Pia and I'm from California.
I'm Molly and I'm from Maine.
I'm Thomas from Louisiana.
Hello, I'm Serena and I'm from Missouri.
And I'm Divya and I'm from Michigan.
This is our team's mission patch. It shows the moon because that is the location of our mission, and the area we drew is Shackleton Crater, a potential landing site for the Artemis program. The sun represents the solar power collection that powers LEMUR. Each of the 7 sun rays represents a different team member, including our mentor, Miss Hanover, and our last names are all written along the bottom. In the center of the crater sits a toboggan with a lemur tail, because our project name is LEMUR, which represents the pulley system that pulls the toboggan in and out of the crater.
Let's start with some background information. In situ resource utilization, or ISRU, refers to using materials found on-site rather than importing them from external locations, in this case being the Earth. This approach is important because of the high cost of launching materials to space. Currently, sending 1 pound of material to the moon costs about $10,000. By utilizing lunar resources, we can significantly reduce these costs and the dependency on supplies from Earth. 2 of the most abundant resources on the moon are ice and regolith. Ice is found in great amounts in permanently shattered regions and can be processed to provide resources essential for supporting human life. Lunar regolith, the loose layer of dust and rock covering the moon's surface, is another vital resource. Not only can it be used for construction and manufacturing, but it also effectively blocks harmful solar radiation, offering protection for habitats and equipment. In this project, we use regolith for manufacturing vehicles. Using its ISRU techniques allows us to harness these local resources, making lunar exploration more sustainable and cost-effective. We aim to explore the southern region of the moon, particularly the permanently shadowed areas of Shackleton Crater at the lunar South Pole, which is of great interest due to its potential ice deposits. This area hasn't been extensively investigated before, but missions like Viper and Artemis are set to explore it soon. Viper, part of NASA's Artemis program, will search for ice and other volatiles in this region. However, Viper could face challenges with sinking into the low-density regolith found in these areas. To address these challenges, our project proposes using a toboggan-type vehicle, similar to a flat-bottom sled, which is more practical for navigating the fine-grained regolith and steeper slopes compared to conventional rovers. This approach is not only suitable for the terrain, but also easier to construct from moon resources, aligning with the goals of Artemis to develop sustainable exploration solutions.
Shackleton Crater has regions of both permanent darkness and almost year-round light. 3 points on the rim of the crater remain in constant sunlight for over 90% of the year. These spots can be utilized for rover resting and recharging through solar energy or future Artemis base camps. The reason that everywhere around the world gets exposed to sunlight during the year is due to Earth's axial tilt of 23.5 degrees. As the Earth orbits the sun, this tilt causes different parts of the Earth to receive varying amounts of sunlight at different times of the year. This axial tilt is responsible for the changing seasons and ensures that over the course of a year, every location on Earth experiences periods of daylight and darkness. The moon has a much smaller axial tilt compared to the Earth, and it has deep craters, some more than 2 miles deep, double the depth of the Grand Canyon. Because of this smaller tilt and deeper craters, some areas of the moon, especially those in the southern pole, are in perpetual darkness. Focusing on the southern pole of the Earth, this region experiences areas of perpetual darkness during the winter months. The Earth's axial tilt prevents sunlight from reaching the southern pole, causing the sun to never rise above the horizon and leading to continuous darkness for several weeks in certain areas. Because of the very steep slopes of the crater's rims being around 30 degrees, typical rovers are not able to traverse down into the permanently shadowed regions of Shackleton. Rather, they would explore smaller, less deep, and wide craters in the surrounding area. For example, the Viper rover, a NASA moon rover, can only traverse slopes of up to plus or minus 15 degrees, which makes it impossible for the rover to observe and traverse the large Shackleton crater. But due to our design using a pulley system and a toboggan-type vehicle, our rover would theoretically be able to travel to the bottom of the crater and explore the potential abundance of water ice and valuable materials in the regolith, such as with metals including iron and aluminum. Because Shackleton Crater is so deep and in permanent darkness, the bottom of the crater reaches temperatures of 40 degrees above absolute zero. In other words, negative 388 degrees Fahrenheit. In order for electrical components to work, they typically need internal temperatures of warmer than negative 58 degrees Fahrenheit, much higher than the temperature of the crater. This calls for the need to maintain a high internal temperature, something that also uses energy. The most productive source of energy on the moon is solar energy, as it is much more efficient in space and on the moon than it is on Earth due to the absence of a thick atmosphere. In order to utilize the best light to energy conversion ratio possible, a solar array located on the rim of the crater in the almost year-round light will be on a tilting axis to be perpendicular to the angle of the sun, normally around 15 degrees. Then, using microwaves, a wireless power transfer to the receiver on the transporter will be completed in order to power tools and keep the electronics warm. Additionally, the solar array system will be equipped with tracking technology to ensure optimal alignment with the sun throughout the lunar day. This precise alignment is crucial for maximizing energy capture and efficiency. The energy collected will then be stored in high-capacity batteries to provide a consistent power supply, even during periods when the sun is not directly overhead. Furthermore, the implementation of wireless power transfer via microwaves not only reduces the need for large quantities of cables, something very expensive to launch, but also allows for greater flexibility in positioning and mobility of the vehicle. This approach ensures that our vehicle can explore the depths of Shackleton Crater without being tethered, significantly enhancing its operational range and efficiency.
Our mission objectives are to design a device that can travel to the bottom of Shackleton Crater and collect resources, to employ in situ resource utilization techniques to prove whether or not it is possible to use lunar regolith to build structures and other apparatuses, and to investigate ways to use solar energy to power devices on the moon. Now for our brainstorming process. Our proposal for utilizing ISRU techniques on the moon is to create a vehicle similar to a toboggan, with a flat bottom and no wheels, curved ends that point upwards like a canoe, and ample space in the interior for carrying materials and instruments. The toboggan will be a better vehicle for traversing the moon's terrain than a traditional wheeled rover. The issue of low-density regolith causing wheels to sink is combated by the flat bottom of the toboggan, which would smoothly glide over the lunar surface. Furthermore, the high walls of our vehicle protect the instruments and materials inside from the dust being kicked up by the toboggan moving over it. Before moving on to the hands-on experimental part of our project, we discussed how the toboggan would explore craters. We would use a winch to anchor the toboggan to the rim of a crater and slowly lower it using a pulley system. Our vehicle would have various places to attach scientific instruments, measurement devices, and cameras, all of which would be sent to the moon from Earth and be charged on the moon via solar energy. Now I'll explain how we harness ISRU techniques in our proposal. The toboggan itself will be made from a clay-like mixture of lunar regolith and water, both of which are resources already found on the moon. We actually experimented with this to determine what type of regolith and how much water is ideal for the sturdiest vehicle, which we will get to soon. Once molded into the shape of the toboggan, the mixture will be left in the cold shade to harden, and when it is fully solid, the scientific instruments and solar panels can be attached. Now we'll explain our experiments in building prototype toboggans and testing them. We started by testing several different toboggan designs by making them out of scrap paper, and we ended up choosing an origami canoe design. The reason we specifically chose this origami design is because it had a flat bottom and pointed ends, which is exactly what we wanted our toboggan to look like, because this allows it to smoothly traverse across the lunar surface. Also, having an origami design would allow humans on lunar missions to just take some square aluminum sheets with them and then assemble the toboggan mold upon landing on the moon. We then cut out the flat bottoms of some aluminum food trays and used the same origami design to shape them into toboggans as well. The reason we chose to use the aluminum trays for the molds is because they're strong and sturdy, yet still flexible enough to shape into a mold. These molds can be reused a number of times to make multiple regolith toboggans. We made 2 identical versions of this mold which were the same size and mass.
Next, after we had the molds, we started making the actual toboggan prototypes. We used 2 different types of lunar regolith simulant in order to test which one made a stronger toboggan. We used simulants of Mare regolith, which is mainly composed of basalt, and Highlands regolith, which is mainly composed of anorthosite. Both of these types of regolith are present in abundance at our chosen location of the South Pole, although there is slightly more anorthosite. One of the initial observations that we had about the regolith is that the Mare regolith simulant was much coarser, while the Highlands regolith was finer. So for each of the two types of regolith, we mixed regolith with water until the mixture reached a clay-like consistency. This process did take a little bit of trial and error, and after that we poured the mixtures into the molds and shaped the insides to get a canoe-like shape. After that, we put them in the freezer overnight at around 0 degrees Fahrenheit or negative 18 degrees Celsius. We made sure that both toboggan prototypes were about the same mass to ensure that we would be able to accurately test them. Here's a quick time lapse featuring Pia and myself that shows our process of making the toboggans. After we took the toboggans out of the freezer the next day, we proceeded to run a series of tests on them to determine which type of regolith made a stronger toboggan. When we're on the moon, it's important that the toboggan is able to travel across the lunar surface as smoothly as possible, and also be able to efficiently traverse across steep environments, such as craters. So that's why the first test that we ran was a sliding test. In this test, we simulated a slope by placing a tray full of regolith at a 25-degree angle using some tape rolls, which you can see in the photo here. We tested both toboggans by manually sliding them through the slope of regolith and seeing which one slid more easily. The next test we ran was a carrying test, where we placed 3 objects of 3 different masses inside of each toboggan. We had one 75-gram object, which is the smaller measuring tape that you can see at the top of this photo, one 157-gram object, which is the larger measuring tape in the middle, and one 232-gram object, which is the stone at the very bottom. Now we're going to discuss the results of our experiments, as well as our overall conclusions. When we did the sliding test, we found that the toboggan made out of Highlands regolith was much easier to slide than the toboggan made out of Mare regolith. Here you can also see the results of our carrying test. As you can see, the toboggan made out of Mare regolith, which is the black one at the top, crumbled a lot more after the carrying test than the toboggan made out of Highlands regolith, which is the light gray one at the bottom. This is likely because of the fact that if we go back to our initial observations about each type of regolith, the Mare regolith is more coarse in general, and the grains are larger. In contrast, the Highlands regolith is much finer. So this could be why the Highlands toboggan didn't crumble quite as fast. We decided that the Highlands regolith was a better option for building items out of. Firstly, the Highlands toboggan was much easier to slide than the Mare toboggan, and as mentioned earlier, it's really important that the toboggan is able to slide well. And lastly, when we performed the carrying test, the Highlands toboggan did not crumble nearly as much as the Mare toboggan did, which proved that it was much more sturdy and robust.
Then we analyzed the chemical composition of common building materials on Earth and compared them to moon regolith using principal component analysis, k-means clustering, and then calculating the cosine similarity. We found that high-tide basalt was very similar to low-tide basalt, and the next similar building material was brick. On the other hand, Highland regolith was a lot more similar to brick than the other two regoliths, indicating it is a more suitable building material. This may be because of the slight differences in chemical composition. Compared to the basalts, the Highland regolith has more aluminum and less iron. We could extract more specific materials from the regolith as well, such as water, glass, and aluminum, which would help create other crucial items. Once the toboggan is built, it will help us mine and explore the moon's surface. This includes getting resources from the bottom of craters that have never been investigated before. A shovel will be attached to the toboggan to collect the regolith and other moon rock. Furthermore, we will add an ice drill to map water sources, which is very important in ISRU for colonies on the moon. Water is essential for human survival and could be used in electrolysis to separate water into oxygen and rocket fuel. We also could sample other parts of the solid moon rock and figure out what exactly makes up the moon. For that, we would use various spectrometers created by Viper, including a neutron spectrometer to detect hydrogen, a near-infrared spectrometer known as NERVIS, and a mass spectrometer known as MSOLO. This study demonstrates how ISRU can be used to create sturdy apparatuses from hardened regolith, providing an efficient, cost-effective method for developing sophisticated lunar colonies. For future lunar missions, in situ utilization of regolith can be pivotal for building colonies by extracting specific resources. The resources gathered by toboggans can be used to manufacture additional instruments, creating a sustainable and self-sufficient cycle.
We have an environmental responsibility to leave the moon and other places we visit in space the same way we found them. In order to do this, we have to make sure that we're not overmining limited resources in order to keep the composition of these other bodies the same. We also can't leave things from Earth or other planets on the moon that could have the potential to damage the environment there, because introducing certain items, materials, or organisms could have the same effect as invasive species here on Earth. While we tried to keep sustainability in mind throughout our project, it's important to find more sustainable methods in the future. Now we're going to reflect on our time here at SEAS. The SEAS program was a unique experience for all of us and allowed us to learn many new things about STEM from experts in specific fields related to our research and project work. The main challenge we faced was the limited amount of time we were given to complete the project, which led to certain aspects of it feeling rushed. Additionally, there weren't many opportunities for bonding or interacting with our team or members of other teams before we immediately jumped into work, which presented problems with team dynamics. Some of the highlights of this program were meeting and working with like-minded peers from all over the country who are all passionate about space, and speaking with mentors and experts who are knowledgeable about subjects we're interested in, though there are many more. We are very grateful for having the opportunity to participate in the SEAS program because we met many new people our age who are passionate about the same subject we are. It was a great learning experience that taught us not only about STEM, but also about how to work with people we have only just met.
I'm incredibly honored to have been selected as a NASA SEAS intern. This week has been an extraordinary journey, working alongside my amazing teammates and mentors. Meeting astronauts such as Dan Tanney and mission leads like Parker Francis have been an amazing experience. I had the incredible opportunity to network, learn about microgravity, and delve into the intricacies of space exploration. The knowledge and connections I've gained are invaluable, and I'm truly grateful for this unique experience.
I am so grateful to have participated in the SEAS program and furthered my passion for space. I learned so much this week and will always cherish the memories I made here. Thank you to everyone who made this possible.
It's hard to say what my favorite part of the SEAS internship was because I had so many unforgettable experiences. One of the best parts was drawing the mission patch and doing hands-on experiments such as building the boats that helped us better visualize how our project worked.
Being selected for the NASA SEAS internship has been an absolutely great experience. Working on the In Situ resource utilization team has given me hands-on experience with the technology and innovative thinking needed for lunar exploration. The opportunity to collaborate with like-minded peers and learn from industry experts such as those on NASA's Viper rover team has been incredibly inspiring. This experience has not only boosted my confidence in my skills, but has also solidified my passion for aerospace engineering and robotics.
This week at the NASA SEAS internship was incredibly rewarding. I loved meeting brilliant people from across the country, as well as our mentors, SEAS staff, and experts who spoke with us. I especially want to thank my team for their diverse perspectives and working styles. I also want to thank the mentors who woke up early to go to the gym with us. It was very interesting to learn about the moon and the Artemis mission, and this experience as a whole was awesome.
I am so unimaginably grateful to have had the opportunity to be a NASA SEAS intern. One of the things that I think is the most unique about this program is its emphasis on real-world application of scientific principles. After working with my team and mentors on this project, I can definitely say that I've learned more in this short week than I have in any other week in my entire life.
So many people more experienced than us helped along the way as we navigated through each stage of SEAS and developed Lemur into what it is now. Stephanie Hanover, our mentor, supported us every step of the way as well as facilitated team bonding, and we are so grateful to her for all that she's done for us. We would also like to thank Laura Tomlin, Selena Miller, and Margaret Baggio, as well as the rest of the NASA SEAS team for coordinating everything, keeping us safe for the week, and assisting us with our project as well. Zoe Zeladik, a SEAS alumni, gave us a lot of tips as well about this project, the ZQube and parabolic flight. Additionally, Parker Francis, a Viper rover team member and driver, spoke with us first online and later in person about our project, since the Viper and Lemur missions overlap in many ways. He gave us so much helpful information and ideas that really helped shape our project and steer it in the right direction. Jim Heisey, an engineer at Iowa State University, also talked with us later on in our project and helped us make our last few tweaks to Lemur and finalize the design. In addition, we have many other speakers who provided insight on various space-related topics, including Bob Twiggs, Dr. Kevin Crosby, and Dr. George Pantelos. Finally, here are our works cited. Now we'll open it up for any questions.
Excellent work. We invite the In Situ team to please turn on their cameras and unmute their microphones. We have several questions for your group. What problematic aspects of your design would need to be addressed before implementing this design on the moon?
Yeah, I'll answer that one. So, our design, it should be used with in a future aspect where people are already on the moon, because it needs to be built without robotics with people as well. So that's one of the big aspects that it needs to be further along where there's people already on the moon.
And the next question for your team is, why was Shackleton Crater chosen as the location?
Yes, I can answer this one. So Shackleton is really important because it's already like future missions have classified that there is ice water in the deep in like the bottom of Shackleton Crater, and Artemis and Viper they are already planning to go to Shackleton Crater, so this would be a great complement to both of those missions.
And your next question, where does the water come from? Do you bring the water from Earth?
I can answer this one. So as you know, our toboggan will be largely made out of a clay mixture of regolith and water, and one of its main purposes is to go to the bottom of craters to extract ice water. So for the very first toboggan that we make, we won't have the ice yet because we wouldn't even have gone into the crater yet. So for that, we would have to use water imported from Earth to create that toboggan. However, after we use that toboggan to go down craters and extract ice, we can then melt that ice that we get and use that to make more additional toboggans. And we can also use it for several different purposes, such as drinking and energy.
How did you come up with the pulley design system for the rover?
Yeah, I'll answer that one as well. So, because of solar winds hits the moon really a lot more than it can hit the Earth because of our magnetic fields, and solar wind is made of positive ions and electric and negative electrons. And because the electrons are faster and lighter than the ions, they hit the surface much faster and make it more electromagnetically charged. So because it's electromagnetically charged, there's more friction on the moon. So when trying to slide something down the surface of the moon, because there's a lot more friction and it doesn't slide as well. So we needed a force to act upon it to move it all the way down.
And then when recommending this internship to your peers, what would you offer as advice for future applicants?
Yeah, so see, it's a really great opportunity, and you get to talk with so many knowledgeable people about space and more specifically whatever you're researching. So maybe like the biggest advice is to ask questions. There's multiple speaker talks which were really helpful in the design of our mission, and yeah, it's just really informative.
So there's a question about how would we get to the moon without a devastating waste of environmental resources, let alone bringing the materials to build an entire base there?
So that is why we are experimenting with in situ resource utilization techniques, so that we're utilizing resources already on the moon, so we don't waste a lot of money and resources bringing things from Earth to the moon. So using resources already on the moon, while it will still take things from Earth to build an entire base there, we're trying to use more of like regolith, ice, and whatever else is found on the moon.
Did you consider using 3D printing as a possibility for developing your toboggan system?
Oh yeah, for sure. 3D printing could be used on the moon because we would still be using the regolith and the ice water to make the mixture, but then 3D printing could definitely be used as a way to create our toboggans. And also the infrastructure for the large 3D printers could be made out of aluminum that's also extracted from the regolith.
And what was the most surprising part of the internship to you?
I can take that one. So one of the most surprising parts was the amount of people I've met and like the connections I've gotten, because I never thought I'd meet so many other people who love technology and engineering like as much as me, but this experience has really allowed me to expand my network and like make friends across the country.
Excellent. So you did a wonderful job on your research and congratulations to your team. Our next project is the In-Space Plant Growth Systems led by Laura Tomlin. Ms. Tomlin is the Senior Outreach Coordinator at the University of Texas Center for Space Research, where she works to bring STEM and space opportunities to students and educators. Laura is dedicated to the idea that real research is the best way to increase student engagement in STEM. Please join me in welcoming the In-Space Plant Growth Systems team with their project Cultivating a Galactic Garden.
Hello everyone, my name is Daniel Casares and I am a part of the Plants in Space group at SEAS, and this is our presentation, Cultivating a Galactic Garden.
Hello, my name is Ashley Krieger and I'm from Arlington, Virginia.
Hi, my name's Liz and I'm from Rogers, Arkansas.
Hi, my name is Isaac Hildebrand and I'm from Salt Lake City, Utah.
Hi, my name is Daniel Casares and I am from Chaparral, New Mexico.
Hi, my name is Rebecca Humphreys and I'm from Tucson, Arizona.
Hi, my name is Rowan Hankins and I'm from Salt Lake City, Utah.
The presentation is split up into 4 main sections: the background, the Hitchhiker Seed Project, the ZQube project, and the conclusion.
Plants play a crucial role in the future of space missions, both as a sustainable food source for astronauts and as an important resource if humanity ever hopes to establish life on the moon and, eventually, Mars. Changes in gravity, both brief and prolonged, place an immense amount of stress on plants. For example, stress can cause disturbances in the calcium ion distribution in plants, which is important for signaling, membrane stability, and maintaining cell wall structure. This distribution weakens the cell walls and overall structure of the plant, making them more susceptible to damage, like stunted plant growth. In the absence of gravity, another stress adaptation from plants is the loss of direction perception, resulting in erratic growth patterns. Additionally, lunar regolith, or lunar soil, is highly hydrophobic in nature. This nature negatively impacts the plant's absorption of water and nutrients, further compromising its health and development. To better understand the effects of microgravity on plants, radish seeds that were previously flown on a parabolic plane were planted in 6 different sample containers, each consisting of 9 seeds. Half of the sample Petri containers were flipped upside down twice a day, once in the morning and once in the evening. Their irregular growths demonstrated the abnormalities caused by exposure to varying gravity. The results showed that the spun seeds germinated at the same rate, but the roots formed a loop around the center of the seed.
The composition of lunar soil is drastically different from Earth soil because of the moon's airless body that doesn't have the benefit of geological processes of water and wind erosion. Its high composition of mineral and lunar rock fragments phobic properties. Irrigation and soil management techniques. Based off of the ISS Veggie and Advanced Plant Habitat Systems, exploring long-term effects of microgravity and identifying patterns of plant growth will help analyze seed germination failure. Studying plant patterns in space leads to new insights into their biology and stress responses. Our mission is to pioneer sustainable agriculture in lunar regolith by drawing from ISS methods, and we aim to address the unique challenges of water movement as well as nutrient retention in lunar soil.
The Hitchhiker Seed Projects. We will send Arabidopsis seeds and seedling samples up in a parabolic flight. These little seeds hitch a ride with other experimental payloads on the aircraft and thus are referred to as hitchhikers. So, what will we be flying? We will fly 2 samples, seeds and seedlings. Arabidopsis seeds will be placed in an airtight pouch. The seedlings will be placed in water agar, which will grow 3 days prior to the flight. There will also be seeds from the same batch kept on the ground and grown at the same time and under the same conditions, other than the microgravity. The seeds kept on the ground will be used as a control group to isolate exposure effects. Effects of microgravity on Arabidopsis growth. Once the seeds get back from their flight, they will be grown and studied for long-term effects. The factors that will be observed include root growth, plant health, and how temperature can affect growth of the exposed seeds. Utilizing the translucence of the agar, the pattern and length of Arabidopsis roots will be observed. Previous NASA studies have recorded erratic root growth and direction. Pictures will be taken of root patterns and compared to a color chart and a root chart to monitor the plant's calcium levels and effective root development. Root development is a critical phenomenon in growing plants in varying gravity because it is necessary for proper growth. The reason plant health is a huge topic to look into is due to the fact that plants experience stress in microgravity, which usually leads to abnormal or stunted growth. Leaf size is also essential for growing plants on the moon because without large, good quality leaves, plants are inedible. The seeds will be planted in small pots and moved in environments with different temperatures. The first group will be placed under a heat lamp at 40 degrees Celsius. The second trial will be placed in a fridge at 4.4 degrees Celsius. The third and final trial will be kept in a temperature-regulated room at 22.5 degrees Celsius. The purpose to observe extreme temperature tolerance of exposed seeds. This is very important because the moon has extreme climate conditions, with temperatures going all the way up to 127 degrees Celsius and getting as low as negative 250. The different environment will help us understand how exposed seeds compare to regular seeds in varying climates. To monitor seed temperature tolerance accurately, there will also be a control group of seeds that have not been exposed to varying gravity, under the same conditions in order to isolate the effects of microgravity on the exposed seeds. During the flight and before starting the growth cycles, the seeds will also be kept in a temperature-controlled room with conditions of 22.5 degrees Celsius.
Our next slide will be about logging our growth. In order to maintain the growth of our seed samples, we will each keep each other accountable by keeping a data and watering log. In addition, there will be 3 repetitions of growth cycles to make sure the data provided is accurate. Each of the 3 repetitions will be flown and exposed to microgravity in a parabolic flight and will each be placed in different agar samples. And we will also be using Arabidopsis. This was because a huge benefit is that Arabidopsis has a lot of studies that we can compare our data that we find to. We will also have a separate control group to monitor, that way we can make sure we record and log all changes done when exposed to the different factors, like the temperature. We will also be recording and logging data in our schools for the most part. This is because we want to educate our local communities in our respective areas in order to educate about the importance of understanding microgravity and its effects.
Next up, we are going to talk about our experiment with water diffusion through regolith using the ZQube. One issue that will be a problem when it comes to growing plants on the moon using lunar regolith is the fact that, as was said earlier, lunar regolith is very fine and very compact. This makes the lunar regolith very hydrophobic, and it makes it very difficult for capillary action to occur, which means that water cannot diffuse through the lunar regolith. It is important that this issue is overcome, as plants grown in lunar colonies will have to be grown in lunar regolith, considering that it is very abundant on the moon. There are several different types of lunar regolith depending on what region of the moon you are on, but for this experiment we used a Highlands lunar regolith simulant from NASA STEM Materials. To help us conduct this experiment, we are using the ZQube, which is a prototype microgravity sensor developed by Twig Space Labs. As seen in the graph on the left, the ZQube contains an LED light at the top, a spot for an experimental payload, a sensor stack, and that sensor includes a camera. The sensor stack also includes an accelerometer, humidity and pressure sensors, temperature, and a magnetometer. And we will be putting a payload into the ZQube for a parabolic flight. Okay, so the goal for the water diffusion through regolith project is to learn more about potential water diffusion techniques for lunar regolith. The primary measurement that we will be taking is the moisture level of the regolith simulant. The variables in this project, independent, will be the microgravity environment, as well as the hypergravity and regular gravity, as well as everything in between during the parabolic flight, as well as whether or not perlite is useful in assisting water diffusion through lunar regolith. The dependent variable in this project will be how well the water is absorbed and diffuses through the regolith simulant.
Pre-work. Before reaching our final product, many experimental procedures were held to discover the materials needed and the methods to perform these by. With the goal of this project in mind, we utilized a wick, perlite, and Orbeez. After a successful drop test where we each experienced microgravity, these were our findings. The wick spreads water efficiently, but not throughout the entire sample of the regolith substrate. Perlite took an extended period of time, but proved to be more thorough in means of range. Finally, the Orbeez absorbed the water before reaching the regolith. The purpose of the additives are to space out the fine particles within the substrate. This will create a less dense environment for the water to flow through, resulting in more efficient capillary action. We decided on perlite as the most effective additive for this specific experiment. Design development. In the experimental stages of this project, we started out with a simple design featuring a tube filled with lunar regolith substrate and tilted at an angle for optimal video angle capture from the ZQube. The water would be added right on top before takeoff. For our second design, we added wicking and perlite to the lunar regolith substrate, testing the effectiveness of additives. For our next design, we took out the wick, figuring that there were too many variables involved with the testing. The issues experienced in the first couple of designs mainly revolved around the water mixing with the regolith substrate samples before ever experiencing the microgravity. Final design. These models represent the experimental structure of our final project. We first separate the container with a tube and then create a secure barrier with hot glue. On each side, we sampled lunar regolith substrate. To one of those sides, we added perlite. Right before the beginning of the experiment, water will be added to the tube in the sensor. The idea is that during the microgravity, the water will begin to crawl up the sides of the tube and out onto the surrounding samples. After the variations of gravity have been experienced, we will observe the movement of the water using the camera on the ZQube. We will also use the needed measurements on the sensor stack, including humidity.
This slide contains the procedures that will be carried out during the parabolic flight and in the hangar beforehand. The first step will be placing enough lunar regolith simulant on the left side of the barrier and well to cover the bottom of the lid. After that, equal parts lunar regolith simulant and perlite will be mixed together, and for step 3, that mixture from step 2 will be placed on the right side of the buried well enough to cover the bottom. Next up, the center well will be filled 3/4 of the way with dyed water, and the water is dyed so that it's easier to see when the water leaves the well from the camera. Finally, the experimental payload will be placed upside down in the ZQube 2 stack, and the light and camera will be started. Our data analysis of this project will be checking the camera footage and looking for darkening of the regolith simulant, which will indicate the diffusion of water through it.
Up next is our conclusion. For our conclusion, we will quickly skim over and summarize both of our project data. Up next is the conclusion, the Arabidopsis. The Arabidopsis growth and development project seedings will be sent into a microgravity parabolic flight, as previously mentioned. The Arabidopsis seeds will then be distributed to each team member's schools with their respective experiments conducted in a classroom setting in different temperatures, and the growth and data will be recorded afterwards. Then, monthly meetings will be held between team members to discuss all of our findings, and our goal is to better understand the effects of microgravity on plants. Data will be shared between classrooms, resulting in a national collaborative experience.
Utilizing the ZQube's tools, we will monitor our payload, which will hold lunar regolith simulants, one with perlite and the other without. They will be placed in a microgravity environment in order to further understand how water and regolith interact in a microgravity setting. We would like to extend a special thank you to the following people. Laura Tomlin, our wonderful project mentor who aided in our research and agreed to carry our hitchhiking seedlings on the parabolic flight. Margaret Baggio, the SEAS Program Director. Selena Miller, the SEAS Program Manager. Jacob Torres, who provided an immense amount of information and took the time to answer our many questions. Dr. Mickey Sodi, who met with us virtually and contributed to our initial research. Becky Fritchie, the best van driver we could ever ask for. And all of the other SEAS Program members who we couldn't have done this without. Thank you for your time today, and we will now be taking any questions that you may wish to ask.
Awesome. We invite the in-space plant growth team to please turn on their cameras and unmute their microphones. We have several questions for your team. The first question is, on the seed germination experiment, is the root growth a result of being flown in microgravity, or are you modeling the effect of microgravity on the roots?
So I can take that one. And so for the growth of the seeds, the way they grow in a circle, that was because we turned them and modeling the effect. But there are still a number of factors that being exposed to microgravity affected. For example, the metabolism is slowed, and the overall health of the plant isn't as good as like a normal plant. Like if they absorb nitrogen, it doesn't go out as quickly. And if we didn't do the modeling, like with the flipping, the roots would have grown, like they wouldn't grow the same as a normal plant. They wouldn't grow in a circle though. They'd just like be smaller and not as in depth. So
great.
It was a result.
Thank you. What made you choose to focus on growing plants on the moon instead of on Mars?
Okay, yeah, I can take this one. We chose the moon just for the sole reason that that's coming up first. Mars is obviously considerably farther away, and so we decided to work on focusing on growing plants on the moon because it's going to be a lot sooner to have people spending extended periods of time on the moon compared to Mars, because of how close it is. Obviously we already have the Artemis missions going, and within the next couple of years, there will be more people on Mars again. So plants on the moon is definitely going to be the first one to be an issue compared to Mars.
How did you use the growth experiments currently on the ISS to guide your research?
Office is closed.
Anybody on the team?
Yeah, I can take that one. So when we were looking at the ISS and like different soils to do it in, that on the ISS they used the advanced plant habitat, and they use like veggie soil. And so when we were looking at different soils to use, we initially thought that we should use that one, but we wanted to be able to use resources that were in situ, like on the moon, because we wanted to grow plants on the moon. So when we were looking into that, that was how we helped examine other soils and different plants to grow, because they have done a lot of experiments with Arabidopsis. So that's how we also picked the plant.
Okay. What were your goals for the experiment? Would it help with future space exploration, or were you just focusing on a lunar habitat?
So I can take this one. It is, of course, going to help with the lunar habitat because it's not really viable to take soil up to the moon over and over again every time you want to plant something. So it'll 100% help with cultivating the moon. But additionally, we have experiments going for just microgravity in general and for changing gravity. And for anywhere you go in space, whether it's Mars, whether it's space stations, wherever it is, you're going to have
changes in gravity that you're going to have to know how to overcome and how to move forward with the barriers and obstacles caused by that. So it is going to help with space exploration as a whole.
And how could this potentially benefit people on Earth?
I can take that question. So, the information and data collected, like to help with this experiment on the moon to help grow plants on the moon, this can help people on Earth by like furthering research opportunities and understanding of the processes that are needed to help establish like a lunar habitat on the moon, and just generally help with understanding of plants.
Well, you did an awesome job with your research. Thank you to the team. Our next team is the Microbial Application and Space Group, led by Laura Tomlin, the Senior Program Manager at the Center for Space Research. Please welcome the Microbial Application and Space team with their Project BFF Biofilm Fighters.
I am Olin.
I am Monster Act. I am Dahana.
I am Jin.
I'm Valeria.
We
are BFFs.
Hello, we are biofilm fighters from Project BFF. Our project revolves around combating microbial contamination in space, evaluating the efficacy of oleic acid, and inhibiting biofilm formation by Pseudomonas florensis under microgravity conditions.
To give a brief overview of what you can expect to see in this presentation, we have split it up into 7 portions. The first portion is the mission statement, which will be a brief overview of our project's goals and research questions we asked. The second portion is the overview. We will cover the theory and concepts of biofilm, oleic acid, and other materials used, as well as preparation tactics and research processes. The third section is the expected results, which will include our hypothesis, concepts, and theories. The mission requirements section will include a full list of mission requirements, as well as a list in condensed form. It will also include the concept of operations. The design overview section will include design, processes, and procedures, as well as compliance checks and functional block diagrams. This section will give a complete overview of our ZQube and our plans for microgravity operations. The management section includes team organization and roles, as well as the budget we use to purchase materials. And lastly, the conclusion section includes a summary of our presentation and research, as well as a final slide where you can contact our team members.
To start this presentation, we will review the mission statement. The goal of BFF is to analyze the effects of phytochemicals on biofilm that are exposed to microgravity, aiming to help control contamination on commonly used materials in the International Space Station.
Next is the mission overview. This is a summary of the aims and research of our project. To understand the complex challenges posed by biofilms, it's essential to delve into the fundamental theories and concepts underlying their formation and behavior. Biofilm is a three-dimensional assemblage of microbial cells and microorganisms. These cells are intricately packed and embedded in a matrix of extracellular polymeric substances. Biofilm consists of polysaccharides, proteins, extracellular DNA and RNA, lipids, and various other biomolecules. Together, these components form a protective layer around the microbial community. This protective layer helps microorganisms thrive by safeguarding them in various environments. Constituent cells produce and accumulate material, contributing to the biofilm's resilience and stability. Initially, it was believed that microorganisms only existed as planktonic or floating cells. This view changed in the 1970s when researchers observed microorganisms forming biofilms in aquatic environments. Biofilm formation takes place in 4 main steps. The first is reversible adhesion. This is where planktonic cells will attach temporarily. The next is irreversible attachment. This is when the cells form a stable monolayer. Next comes maturation. This is when the biofilm develops 3D structures with polysaccharides. And finally comes dispersion. This is where cells will detach and spread to new areas. Current research is aimed at blocking microbial adhesion and quorum sensing, which is the communication system for the bacteria itself. Microgravity inhibits biofilm formation, reduces metabolism, motility, and proliferation of bacteria. Research aims to develop strategies to control biofilms, which are crucial for medical and space applications.
Oleic acid is a long chain of carboxylic acid. It's a phytochemical. A phytochemical is an organic compound that helps fight off and prevent fungi, bacteria, and plant viruses. The chemical equation of this acid is C18H34 and O2. The acid had two crystalline forms, alpha form and beta form. Alpha form melts at 13.4 Celsius, while beta form melts at 16.3 Celsius. Previous experiments have revealed that oleic acid has anti-biofilm properties. This is done by inhibiting the early stages of biofilm formation by preventing bacteria that creates biofilm, like the P. gingivalis bacteria, from latching onto the surface. And without a surface, the biofilm cannot form.
What other research has been done in the past between oleic acid and biofilm? In an experiment, oleic acid was tested on its antimicrobial properties with biofilm. It was discovered that oleic acid can induce a stress response on gram-positive and gram-negative bacteria present in the biofilm. In this research, a standard crystal violet quantification method was used. The standard crystal violet quantification method revealed that the oleic acid's concentration and biofilm inhibition have a proportional relationship. As the oleic acid's concentration became stronger, the biofilm inhibition was noticeably increasing. Within the biofilm, filamentous biofilm was mostly inhibited, and microcolony formation was completely inhibited. As a result, the thickness and biomass of the biofilm decreased. The polysaccharides and carbohydrates that contribute to the formation of biofilm reduced from 35 to 41% and 26 to 47%, respectively. Lastly, lipase production can also quantitatively measure biofilm inhibition since they promote biofilm growth on a host tissue or material. The biofilm's lipase production decreased between 8 to 30%. In the picture on the right, the first and third rows on figures B and C represent biofilm cultures without oleic acid. The second and fourth rows on figures B and C represent biofilm cultures with oleic acid. From statistics to visuals, the inhibition of biofilm from oleic acid has been prominent. Pseudomonas aeruginosa has a significant presence on the International Space Station, worsening the concern for the possibility of infections on astronauts. In a microgravity environment, Pseudomonas aeruginosa's cell size is smaller than its normal cell size on Earth. On the graph, we can notice that Pseudomonas aeruginosa's biofilm surface area coverage decreases in a microgravity environment. From its boundaries to its biomass, the stress of a microgravity environment has weakened the biofilm formation of Pseudomonas aeruginosa. We hope that oleic acid will become an even more effective solution to combating biofilm formation than its notable performance on Earth. Since Pseudomonas aeruginosa has a biosafety level 2, we decided to use another bacteria to be a model in our research. Pseudomonas aeruginosa is an opportunistic pathogen. If an immune system is low, it is capable of infecting an individual. Pseudomonas fluorescens, the model and a relative of Pseudomonas aeruginosa, is a biosafety level 1 bacteria and can pose less threats in the microgravity environment and research team.
In order for us to attain the biofilm, we need to cultivate Pseudomonas fluorescens. To do this, we will be using the Pseudomonas bacteria cultures kit from Carolina Biological Supply. An aspect of the cultivation process that we can explain is the preparation, which consists of 4 main steps. The first step is the material preparation that consists of obtaining pure culture of Pseudomonas fluorescens and the preparation of 2 vials containing a known quantity of Pseudomonas fluorescens suspension. After this is the microgravity exposure preparation that secures the 0G setup to ensure everything is properly sealed. Next is the post-flight transferization of Pseudomonas fluorescens from both vials to separate petri dishes where oleic acid is evenly distributed on both petri dish surfaces. The last portion of the process is incubation, where the dishes are put into standard conditions conducive to the growth and monitoring of biofilm. The next part of this section, Theory and Concepts, is the method in which we are analyzing our data. We will be using adhesion assay, which is a quantitative method that assesses how well bacteria can adhere to a surface. Although, since there are various ways this method can be used, both quantitative and qualitative data can be collected. The specific type of adhesion assay we are using is crystal violet quantification, which provides information on the mass of the biofilm. This method uses two main components, the decolorizing solution and the spectrometer. The decolorizing solution is added to a volume equal to the culture media, then is placed in an incubator, leaving only the crystal violet infused solution. After this, the spectrometer is used to measure the range of crystal violet absorbed by the bacteria. This method is key to our research because the crystal violet solution absorbed by the bacteria is proportional to the number of cells in the biofilm. The specific procedure starts off with 100 microliters of media, 10 microliters of inoculum being added onto a microplate, then incubated for 24 hours. The next day, the plate is washed in water, leaving only the biofilm attached to the microplate. After the microplate is fully dried, 1% crystal violet solution is added and incubated for 15 to 20 minutes. Following this, the excess dye will be removed with water, then set to air dry for 15 minutes. Next, the recoloring agent is added at a volume equal to the culture media and incubated for 10 to 30 minutes. After this, the crystal violet infused decoloring solution will be transferred to a clean plate with blanks of decolorizing solution. Lastly, the absorbance of crystal violet will be assessed through a spectrometer.
Our third section is expected results. We predict that the P. florensis, when exposed to microgravity, will become more susceptible to oleic acid, which prevents the spread and growth of biofilm. We expect to be able to observe a visible and quantitative difference between the growth of P. florensis exposed to microgravity and the P. florensis that stays on Earth through a microscope and crystal violet quantification, which is measured in micrograms per milliliter. Concepts and theories. This is anticipated because of previous research done on biofilm in microgravity. According to a research article by Pamela Flores from Bioserve Space Technologies and other researchers, the growth of biofilm is weakened when exposed to microgravity. Another piece of evidence that supports our theory is that when some organisms are exposed to an environment that causes strain, it can become weaker. An example of that is our own immune systems. Say after a hard week of exams or work or even school, we could become sick due to previous stress and strain on our bodies the week before. The effects. If these results are reached, then this could be a way to prevent the formation of biofilm on the ISS, specifically the produce on the ISS, because that's not only harmless for astronauts and plants, but it prevents plant viruses as well. Finally, oleic acid is a safe way to clean materials and tools set up on the ISS, and that are already on the ISS in a way that would not have to risk the health of the astronauts. That is how these results could have an effect.
Our next objective are mission requirements. This refers to the detailed specifications and constraints that a mission must meet to achieve its objectives. To successfully carry out our investigation into biofilm formation under microgravity conditions, several critical mission requirements must be met to ensure accurate data collection and analysis. Here are some of the objectives. Microgravity exposure. Expose Pseudomonas florensis to microgravity conditions. Post-exposure treatment. Apply oleic acid to Pseudomonas florensis after microgravity exposure. Biofilm measurement. Measure the extent of biofilm formation after treatment with oleic acid. Comparative analysis. Compare biofilm formation in treated microgravity exposed samples with treated non-exposed samples. We also have some system requirements for our project. Here are the sensors that we will need for our project to be a success. We will need to use the temperature sensor, humidity sensor, pressure sensor, and elevation sensor within the ZQube. Besides that, we will need the following equipment for success. Devices for crystal violet quantification, culturing apparatus for Pseudomonas florensis, incubator for maintaining optimal temperature and environmental conditions, pipettes or automated dispensers for consistent oleic acid application, system for collecting, storing, and analyzing biofilm formation data, and software to process and compare data between treated and untreated samples. This experiment aims to understand the effects of microgravity and oleic acid on biofilm formation, which is crucial for maintaining hygiene and safety in space environments. For this project to be considered a success, there are minimum requirements that have to be met. For minimum data collection, there are two requirements. Successfully expose Pseudomonas florensis to microgravity and treat with oleic acid. Obtain at least one set of biofilm formation measurements for both exposed and non-exposed samples using incubation and formation techniques. There's only one requirement for minimum data analysis: the ability to compare biofilm formation between exposed and unexposed samples. The expected results for this experiment are to observe one of the following outcomes. Microgravity exposure does not affect overall biofilm formation. Microgravity exposure increases biofilm formation, and microgravity exposure limits biofilm formation. All outcomes will indicate the effectiveness of oleic acid in preventing biofilm formation under microgravity conditions.
Overall, our project wishes to utilize biofilm formed with Pseudomonas florensis exposed to microgravity to evaluate how effective oleic acid is in preventing the formation of biofilm. Our project utilizes sensors on the ZQube to collect data that will be stored in the ZQube's SD card. For minimum success, we will measure the effectiveness of oleic acid in preventing biofilm by quantifying and observing changes using crystal violet staining. Overall, our project wishes to utilize biofilm formed with Pseudomonas florensis exposed to microgravity to evaluate how effective oleic acid is in preventing the formation of biofilm. Our project utilizes sensors on the ZQube to collect data that will be stored in the ZQube's SD card. For minimum success, we will measure the effectiveness of oleic acid in preventing biofilm by quantifying and observing changes using crystal violet staining.
Section 5 will be the design overview. This section includes a summary of design elements and principles that we include in our project, including experimental processes, diagrams, a basic payload layout, and a check to ensure compliance. Section 5 will be the design overview. This section includes a summary of design elements and principles that we include in our project, including experimental processes, diagrams, a basic payload layout, and a check to ensure compliance.
The diagram on the left is of the controlled biofilm, Pseudomonas florensis, formed on Earth. We placed the biofilm in a petri dish and magnified it to see the amount of bacteria present. In the middle diagram, we covered the biofilm with a phytochemical called oleic acid. Based on past experiments conducted on Earth, we predict that the oleic acid will encase the biofilm, preventing it from further growth, as represented here in the magnifying glass. The third diagram to the right is the exact same procedure as the one in the middle, except the biofilm cultivated on the petri dish was exposed to microgravity. Once we receive the biofilm back on Earth, it will be coated with oleic acid. Our hypothesis is that the biofilm will weaken when exposed to microgravity, increasing its reaction to the oleic acid. Our predicted results are shown in the magnifying glass that is tinted by the oleic acid coating, showing the decrease in the amount of biofilm.
The ZQube contains an LED light, payload, camera, sensors, and an SD card. The payload will contain a biofilm culture of Pseudomonas florensis. We will measure within the ZQube for temperature, humidity, pressure, and elevation.
Our project utilizes the ZQube payload, which is 3 centimeters in diameter times 3 centimeters in height. Our materials consist of the ZQube, 5 fluid ounces of oleic acid, crystal violet, ACS grade, and P fluorescence. As for hazard prevention and safety, oleic acid is common and poses no threat to humans unless pre-existing health conditions are present. Pseudomonas florescens will be used because of P. aeruginosa's status as a biosafety level 2 bacteria. As previously mentioned, the ZQube will record data on humidity, temperature, pressure, and elevation. As for project deadlines, this final recording is being submitted on Friday, August 2nd, 2024.
Next, we will be speaking about how we managed our team to set us up for a successful project in flight. This section will include team organization and roles and the budget we used to purchase materials. As you can see, we developed a team organization chart which allowed each team member to work on what they specialized in while allowing input from our team's mentors. Our team's mentors oversaw our project and offered valuable tips and knowledge on how it could be improved. Each team member took on relatively equal amounts of work, so nobody became too stressed out due to either workload or time constraints. This slide shows our full budget and a complete list of materials purchased over the week. We were able to stay within the budget by choosing to analyze all product purchases and researching their costs before choosing to implement them. Also note that if you wish to contact our team's mentors and helpers, their info will be at the bottom of this slide.
To conclude, Biofilm Fighters wishes to compare the effectiveness of oleic acid on a biofilm sample that is exposed to microgravity and one that is not. This research will be submitted to the American Geophysical Union and the ISS International Lab to use our findings for a safer environment aboard the ISS. Further research will include testing different antimicrobials to find the most safe and sustainable method for biofilm prevention.
So we invite all members of the Microbial Applications and Space team to turn on their cameras and unmute their microphones. Our first question is, how familiar were you with the topic of biofilm prior to this project? And what made you choose biofilms as opposed to other microbial challenges in space?
So I'm going to go ahead and take this one specifically. Coming into this project, a lot of us had a foundational understanding of what biofilm was, but it wasn't on a deeper, we didn't have an understanding of it on a deeper level. We had an understanding of their structure and their impact in various environments, but we didn't have a lot of understanding of their actual applications. The reason that we decided to choose biofilm itself is because they present a particularly formidable challenge in the unique environment of space. They're notoriously difficult to eradicate due to their protective matrix layer over the microorganisms themselves, and they can lead to significant operational issues on spacecrafts, including malfunctions and health problems. So we decided to choose biofilm because not only was it something that we had kind of a foundational understanding of, but because of the application that they can apply, the application of learning about them can apply to on the ISS and space environments themselves.
So the next question for your team is, what would you consider to be the most difficult part in completing your project and collecting data?
I'm going to go ahead and take this one as well. And specifically, one of the most difficult parts that came up with collecting our data and coming up with a project was not only deducing what type of bacteria to use. So originally, our first idea was to use Pseudomonas aeruginosa as our bacteria to conduct this experiment. However, that presents a level 2 biosafety hazard. So we essentially had to deduce which bacteria was easiest to use and was a safe thing that we could use in our experiment, which ended up being Pseudomonas florensis. And on top of that, in completing the project itself, it's being very careful with the incubation process, and it's being very, very particular in the processes that we take to ensure when we get that biofilm back on Earth, we're taking the process to ensure that we are forming it right and we are getting the data to adequately complete the experiment.
And what complications do you feel might arise with your results?
I'll take that one. So a big part of our process for the experiment was figuring out what method we should use. And when we decided on crystal violet quantification, what we looked at was the disadvantages and the advantages of that specific method. So the disadvantages were that we have to be very particular in the variables such as the incubation and the temperature that the biofilm will reside in. So that's something that we have to look in and explore a lot further and make sure that we are very exact in our method.
And your team seems to have a really cultivated and great culture. How was forming a collaborative team important to the success of your project?
Hi, I will answer that question. We are all very interested in microgravity and microbiology. So forming a collaborative environment wasn't very difficult for us since we were also enthusiastic into researching our project. I think that we were all pretty enthusiastic. There were really no stressful times or arguments. We all recited it like pretty quickly, and I think we all learned that communication really is a very great technique to have when we are doing a team project.
Great. So thank you to all your team. Great work. Our next presentation is from the Propellant Management Technologies. Their mentor is Mauro Esquivel, a mathematics teacher for Edinburgh Consolidated Independent School District. During his 16 years of teaching, he has also been a professor with the University of Texas Pan American, which is now UT-RGV, as well as with the University of Phoenix, focused on STEM education with at-risk students. This is Mr. Esquivel's first year as a project assistant for the SEAS program. Please welcome the Propellant Management Technologies team.
Hello. We appreciate the opportunity to share our research with others. The project we are presenting today regards propellant management technologies. Hi, my name is Will, and I'm from Iowa.
I'm Aria, and I'm from Basking Ridge, New Jersey.
Hi, my name is Aya, and I'm from Maryland.
Hi, I'm LJ, and I'm from Woodsville, New Hampshire.
Hi, my name is Yulmari, I'm from Juncos, Puerto Rico.
Hi, my name is Juan, and I'm from Vega Baja, Puerto Rico.
My name is Oliver Slade, and I am from Sydney, Montana.
Hi, I'm August, and I'm from Seattle, Washington.
To begin, we will introduce you to our primary objective. The goal of this experiment is to test and analyze the effectiveness of a hexagonal grid barrier propellant management device in limiting fuel slosh, separating liquid and gas phases, and minimizing the forming of ullage in the propellant tank in a microgravity environment through use of the ZQube.
This objective was created with the intention to improve upon existing propellant management devices, or PMDs, in order to solve the following problems that pose threats of engine failure. 1. Fuel slosh poses a major threat to spacecraft's stability and navigational ability. The sheer mass of liquid fuel, combined with constant shifting of the spacecraft's center of mass due to the movement of fluid, can be detrimental to navigational systems and ideal flight paths. 2. The inability to separate liquid and gas phases within the fuel tank also poses a threat, as successful combustion in microgravity relies on controlling the positioning of ullage, or empty space within the fuel tank, to ensure liquid alone moves into the combustion chamber. And finally, in microgravity, fuel adheres to the surface of the fuel tank wall, and gas is trapped by a layer of liquid. This makes the process of venting gas without the release of valuable liquid fuel unfeasible.
There are numerous existing solutions for the problems just outlined, namely PMDs. All these PMDs are flawed in some manner, whether they are heavy, inefficient, or both. They all fall short of ideal solutions. There are three main existing types of PMDs. Non-capillary PMDs, also called positive expulsion devices, partial communication PMDs, and full communication PMDs.
A positive expulsion device, such as a bladder or piston, works by keeping fuel stationary within the fuel tank. As fuel depletes, the device expulses and pressure is maintained using an external gas inlet. Positive expulsion devices excel in separating liquid and gas phases and minimizing fuel slosh. However, the mass of the diaphragm enclosing the liquid can often parallel the mass of the fuel tank. This makes positive expulsion devices unviable in medium to large scale scenarios. Partial communication PMDs are typically lighter and smaller, but typically less effective at fulfilling all necessary requirements for a standalone PMD, and are unable to be used in large scales because they are unable to deliver enough fuel to the engine to sustain flow. For this reason, partial communication PMDs are only able to be used in conjunction with other propellant management solutions.
Full communication PMDs are typically larger and heavier than partial communication PMDs, but are better at minimizing fuel slosh and delivering phase-separated fuel to the engine. These are often viable for standalone use, but each PMD has its own drawbacks and benefits. For example, there is a full communication PMD called a vane. Vanes are linings on the internal surface of the fuel tank. They utilize the natural movement and adhesion of liquid in microgravity to guide fuel to the engine outlet. Vanes are extremely efficient in minimizing fuel slosh, and their low mass makes them a strong option. But vanes are unable to effectively separate liquid and gas when flow demand is amplified, which makes standalone vanes ineffective. Some much heavier PMDs, such as sponges, are capable of fulfilling all requirements at the cost of more mass and more volume.
We are utilizing zero-G's parabolic aircraft to run the experiment in microgravity environment. This flight will allow us several periods of about 22 seconds of zero gravity, allowing for multiple opportunities for the liquid to settle and be observed, meaning there will be several trials of our proposed solution.
To gather all of the data necessary for the experiment, we have been given a ZQube. The ZQube is a handheld laboratory, fitted with Arduino sensors, a small camera, and a payload container. It is designed to run small experiments in a microgravity environment. The sensors we utilize on the ZQube are the accelerometer, the gyroscope, and the camera. We utilize the accelerometer to detect a microgravity environment, and the camera to relate visual observations to data from the accelerometer. Using this combination, we will be able to get valuable information on the effectiveness of our proposed PMD. Our proposed PMD design will sit in an acrylic vial with the ZQube. The outside diameter and height of the payload capsule is 35 millimeters, with an inside diameter and height of 26 millimeters.
The contact wetting angle is a very important concept in fluid dynamics. It's defined by the angle of which liquid and solid surfaces meet. The goal of our experiment is to emphasize wettability, which is the ability of liquid to stay on a solid surface, defined by how the adhesion and cohesive forces are balanced out. Smaller, more acute angles effectively increase wettability and result in an easier transport of fluid. As you can see, a 30-degree angle results in high surface energy and contact with the surface, whereas the 75-degree angle has low surface energy. So, we're using hexagonal capillaries, which has 60-degree acute angles to maximize efficiency.
The goal of our experiment is to prevent the unpredictable behavior of fluid in microgravity through the use of a pioneered PMD. There are 3 components to our proposed hexagonal PMD. Shown on the right is a ring scaled to 1/8 the total height of the fuel tank wall. The ring has the same diameter as the tank wall, meaning it encapsulates the entire mid-sectional area. The ring is subdivided into numerous units of hexagons, with each hexagon measuring 1 centimeter at its largest diameter. It's important to note that a small diameter size is necessary to minimize ullage within the hexagonal tubes. This was done in order to induce capillary flow into the hexagons, where liquid is affected much more strongly than gas. The considerations for the capillaries, especially their size and quantity, is influenced by the desire to maximize surface area and minimize volume, as a larger PMD translates to less available and usable fuel. This implies that the largest possible feasible capillary size, 1 centimeter, with the most hydrostatic attraction, is ideal to minimize volume and therefore mass. Additionally, the ring is gradually sloped downwards toward the center to encourage flow above the ring to flow down into the centermost capillary of the hex structure, directing into the second structure, the sump capillary.
The sump capillary serves as the link between the hex structure and the sump, allowing travel from the center to the desired place, which is the bottom and the location of the sump. It relies on simple principles of adhesion and capillary action, which indicate that any fluid exiting the hex structure will travel and adhere to the capillary, directing fuel flow into the sump. The third structure present is a torus shape, placed inside the side of the hex structure farthest from the fuel sump. This serves as a curved interrupter to flow along the sides of the fuel tank, forcing any liquid into the hex structure and repelling liquid from the then unneeded top half of the tank. This simple addition greatly improves the design.
Because materials in an actual cryogenic fuel system are not readily available, we will make the following substitutions. Instead of liquid oxygen or liquid hydrogen, we will use isopropyl alcohol, or PA. This is because the properties of cohesion and surface tension of the two are similar, as method strong forces of cohesion. This is the most important distinction to make in a microgravity environment, in which the forces of adhesion and cohesion become more prominent and observable compared to gravity. Secondly, instead of the proposed insert being constructed of carbon fiber composite coated with polydopamine cupric oxide, our PMD is modeled with the easily acquired PETG 3D printed filament.
We will first analyze as a control the behavior of isopropyl alcohol in microgravity with no hexagonal structure. It is expected that the IPA will move freely around the container, displaying unaffected adhesion, meaning it will climb unpredictably around the walls of the container. This extensively engineered solution should ideally greatly modify the observed fluid motion compared with the control group. A decrease in fluid movement in slosh, a decrease in adhesion to the container wall, and a visible tendency to adhere to the hexagonal structure are expected. The extent to which any of these occur is variable, but some positive results are expected.
This minimum success criteria for the engineered solution is clear data supporting the occurrence of the expected results. A decrease in fluid movement and slosh, a decrease in adhesion to the container wall, and a visible tendency to adhere to the hexagonal structure are expected. Success depends on these occurrences.
In a couple of preliminary tests, it became clear that addition to our PMD would greatly improve the design. In the first job test, where the hex grid was placed vertically, the IPA can be observed adhering to the capillaries nearest to the container wall. This necessitated the implementation of a torus to guide the fluid to the center capillaries. This is another demonstration of the promising potential of the efficacy of this design. This time, the hex grid is placed horizontally. This is also an earlier version of the solution, which visualizes the need for the sump capillary.
This is accelerometer data taken from the drop test showcased in the previous two slides. The moment in which there is a little acceleration due to gravity, therefore the force spikes in acceleration, indicate the purity of our early experimentation. It is, naturally, suboptimal, but the data is sufficient to draw observations from. In the parabolic flight, we will have much purer data, which the onboard accelerometer will observe and validate.
If successful, this proposed solution could serve as a general method for propellant management. We have chosen to use the Space Launch System, or SLS rocket fuel tank, as a model due to its pivotal role in the upcoming Artemis missions. The SLS is engineered to transport the next generation of astronauts to the Moon and Mars. Given the extended duration of these missions, efficient propellant management is crucial for mission success. The SLS fuel tank carries 537,000 gallons of chilled liquid hydrogen, a cryogenic propellant. As a super heavy lift rocket, the SLS fuel tank is over 300 feet tall. Due to its lightweight and specifically engineered capacity to withstand cryogenic fuel tanks, carbon fiber composite with polydopamine cuprous oxide coating is a strong material compatible with liquid hydrogen. However, this material is still experimental and has only thus far been utilized for the purpose of research.
Under the assumption that this PMD fulfills all requirements as expected, the HEX PMD should occupy the same role as a positive expulsion device. The mass of the carbon fiber PMD, scaled to the size of the SLS fuel tank, is 0.065 the mass of the liquid hydrogen. In comparison, the mass of a diaphragm on the SLS fuel tank is 24.5% the mass of the fuel. More specifically, the mass of the PMD is 0.26% the mass of the diaphragm. This shows that compared to traditional positive expulsion devices, the HEX PMD is significantly lighter and takes up less volume.
There are possible design changes worth exploring. Ideally, in the context of capillary actions, angles should be small so that multiple surfaces are in close proximity, and the contact wetting angle is ideal. The issue with smaller shapes and angles is the necessity for more material to fill the space, so this design comes at the cost of efficiency. However, varying the amount of material from the middle to the edge of the tank can increase efficiency. In a future design, we would implement star or triangle shapes that maximize the ability of the contact wetting angle. In addition, we would consider utilizing aluminum or another lightweight yet strong material. Finally, this project could be expanded to be used in conjunction with other methods, such as pressurized systems.
Because we do not have excellent data yet, it is impossible to pass judgment on the solution in these early stages. After the scheduled parabolic flight, we can observe the efficiency of the solution.
This is our mission patch. The slosh on the bottom of the patch represents one of the primary problems that our PMD is designed to solve. The hexagons that are in the center represent the core of our PMD, the hexagonal grid. The flags on the golden stripe symbolize where we are from and the home we found in Texas with the SEAS program. And finally, the constellation of Texas in the sky represents the 8 members of our group.
Special thanks to all that have helped us with this project. We deeply appreciate the support of Laura Tomlin, Selena Miller, Mario Esquivel, Margaret Baggio, Zoe Static, Becky Frecci, Dr. Kevin Crosby, Stephanie Hanover, and the entire SEAS program and the University of Texas at Austin.
NASA SEAS has helped me to meet mentors that are majorly involved in areas in STEM that I am interested in, helping me to look for scholarships and opportunities that will strengthen my future journey into STEM.
NASA SEAS helped me solidify my passion for aerospace and astronomy, which is definitely something I will pursue in the future.
NASA SEAS was a wonderful opportunity that has solidified my passion in aerospace engineering and space research. Talking with professionals who are working on the same issue we are is not only interesting, but also very exciting.
The NASA STEM program has solidified my readiness and confidence in my ability to work in STEM fields such as all types of physics, astronomy, astronautics, and astrobiology.
NASA SEAS has given me an excellent opportunity in different fields of astronomy, physics, and engineering. It has also given me the experience of doing research in an interactive way. Now I'm one step ahead of making my dreams come true.
Thank you to the people at NASA SEAS for providing me with this extraordinary experience. NASA SEAS has solidified my passion for space research and aerospace engineering.
I'm glad I was able to have a great experience at NASA SEAS. I learned many things about the STEM world, including cooperation, and I thank all my mentors and all of the people that made this program possible.
NASA SEAS was an amazing experience that enabled me to grow as a leader, learner, and human being. This program will jumpstart my career into a STEM field and for that I am forever grateful.
Thank you, NASA.
So we want to invite all members of the Propellant Management Technologies team to turn on their cameras and unmute their microphones. We have several questions. Would a combination of technology be possible with PMDs such as vane with hexagonal, or perhaps spacing the walls with hydroponic seal?
Okay, so you can't use veins and the hexagon structure because the veins will hit the hexagonal structure. And as far as the sealant, it wouldn't work because it's hydrophobic sealant, and we're using fuel, not water. So it wouldn't work, or it wouldn't be very efficient.
Why did you use the hexagonal shape and not a circular shape?
Hi, I can answer that one. So a hexagon structure provides more sharp angles, and sharper angles encourages better flow rate and a better return of liquid to remain separated from the ullage, whereas a circle shape would not really provide that.
How did your team decide to research this issue instead of other problems associated with flight and space travel?
So we decided to go with the issue of slosh because of a few reasons. We could apply the laws of adhesion, which was something that we discussed within our first days at SEAS. And additionally, there was previous research from another team led by our mentor, an 11th grade team, where they discussed slosh, and we could build off of what they were doing and expand on their research.
So were you able to test your preliminary design? And if so, did you get results from that?
Yeah, so we did test our design. We 3D printed the honeycomb and we placed it in the vial on the ZQube, and we dropped it from like around 3 stories high, but unfortunately it was only about 1.7 seconds in microgravity, and the camera did have poor resolution, so we weren't able to see just how exactly the propellant, which was the IPA that we used, reacted with the honeycomb. So we did decide that our results were inconclusive.
Will your team get back together to continue working on this research prior to the microgravity flight?
I can take that one. We will not be getting back together to continue working on this research, but there's a lot of ideas we have for the future of our PMD, such as potentially implementing acute angles instead of obtuse angles in the hexagonal grid. Maybe we implement a grid with like triangular shapes. There's a lot of potential with our PMD. So after we get results from parabolic testing, we might meet back up and continue to design and research our PMD.
Great. Excellent work. Our next team is the Space Debris Solutions Group, led by mentor Mauro Esquivel, mathematics teacher at Edinburgh ISD. Please welcome the Space Debris Solutions team and their Sigma project.
Hello everyone, this is the Space Debris team, also known as Project Sigma. My name is Ruhan Singh and I'm from Knoxville, Tennessee.
My name is Aditi Muluganti and I'm from Onalaska, Wisconsin.
Hello, my name is Aliana Chamorro and I'm from Fall River, Massachusetts.
Hi, I'm Ricardo Micheles and I'm from Mission, Texas.
Hi, my name is Garrett Lee and I'm from Overland Park, Kansas.
Hi, I'm Justo Martin and I'm from Chicago, Illinois.
The goal of Project Shooting Interplanetary Garbage, Material Ablation, or SIGMA for short, is to reduce the amount of space debris in Earth's orbit, thus decreasing the dangers for satellite and other spacecraft. A UV laser in low Earth orbit targets and ablates debris between 1 centimeter and 10 centimeters. This ablation causes the object to decelerate, reducing its orbital lifespan and incinerating it in Earth's atmosphere sooner.
But what is space debris? According to NASA, it's any human-made object in orbit about the Earth that no longer serves any useful purpose. This includes derelict boosters and even paint flicks. And since this debris is moving at thousands of miles per hour, any collision can cause massive damage. As more spacecraft are launched into space, the risk of collision increases. Just this past June, a Russian satellite exploded, sending even more space debris into Earth's orbit and forcing astronauts on the ISS to shelter. Monolithic aluminum blocks are used in spacecraft for their lightweight design, but can be penetrated by space debris at the high velocity they travel at, with many traveling at 10 kilometers per second or faster.
Currently, 4 areas of space debris research are being tackled: detection, tracking, characterization, and solutions. Additionally, there are 2 different types of solutions: mitigation and remediation. Mitigation strategies involve preventing, avoiding, or protecting against space debris, while remediation focuses on actively moving the debris. However, most research doesn't cover remediation. Tracking is a main focus for space agencies globally. However, debris smaller than 1 centimeter is currently unable to be tracked, and improved radar detection is required. Though, if satellites are on course to collide with known debris, the satellites will be directed to move out of the way. Another recommended mitigation strategy, PMD, is a preventative measure for spacecraft nearing the end of their service time, forcing them to lower their orbits, thus reducing their risk for damage and to become space debris. Shielding was also mentioned, with multi-layer hybrid walls being used to lessen the damage incurred by collision with debris. For the last of the mitigation strategies, scientists looked at passive methods, primarily reducing the risk of spacecraft explosions via batteries and fuel, thus preventing space debris from forming. In comparison with remediation strategies for cost, it was concluded that remediation is just as effective as mitigation in both short-term outlooks and should be planned for in the long time.
We looked at many different remediation techniques, including a space net and magnetic tethers. However, laser ablation had the highest reduction rate, tied with the ground-based laser, as well as being the cheapest at a total cost of $500,000 per kilogram of debris removed. This made it very easy to settle on ablation using space lasers as our method for deorbiting the debris in this research.
Ablation works through laser irradiation. When the laser is fired, part of the debris material is vaporized, changing the momentum of the debris and sending it back into the atmosphere. The amount of space debris doesn't increase with ablation, and it provides powerful enough pushes to reduce the lifespan of debris by years. However, since it's more powerful, it requires more energy and precision.
First, we looked at the French proposal for a space laser mounted on a satellite. Such a space laser would be effective at deorbiting both large and small debris, and be more affordable for each piece of debris removed, but there would be a high cost to launch and repair the satellites. Another proposal included solar panels and a mirror to target the laser, making it more precise. However, altitude differences would affect the laser's performance, and the mirror would make the spacecraft more fragile.
Overall, our proposal would provide a solution to eliminating 1 to 10 centimeter space debris using remediation. It would require a satellite sturdy enough to house a laser system, solar panels for power, and control mechanisms including stability systems to maintain its orientation, an arm for maximum freedom and accuracy, and a lens to increase preciseness would also be needed. A tracking system would be used to track debris in real time and continue predicting the trajectory of the particles. The minimum success would be a satellite capable of identifying debris and vaporizing its surface to deorbit it, with the altitude of interest being between 200 kilometers and 2,000.
The cube-shaped satellite would hold foldable, multi-junction solar panels on both sides. The excimer laser would be mounted on NASA's robotic servicing arm, which would be placed on top of the satellite. Sensors, radar, and cameras would track debris, while an algorithm working in conjunction with existing European Space Agency models would calculate the angles for when and where to fire the laser as both the satellite and debris orbit the Earth. A high-capacity battery would power the whole system. The satellite would be launched into low Earth orbit with a polar elliptical orbit to maximize the field of view the laser could fire at.
Materials for the satellite include a framework, which would be stainless steel, titanium, and lightweight aluminum, similar to other satellites of this size, minimizing weight while not sacrificing structural integrity. Furthermore, a large lithium-ion battery, minimum 100V, would be required to power the satellite systems and power the laser pulses. This would be powered by the large solar arrays of the craft, located on your bottom left. At minimum, 6 2x3 meter multi-junction and solar panels would be required to keep the systems online. Lastly, the krypton fluoride UV laser mounted on a motorized robotic arm would be required. The full system would cost around $700 million, but could cost up to $1 billion.
Our design features three main instruments and systems. The laser, attached to NASA's robotic servicing arm, or a similar device, has 7 degrees of freedom, which allows for unrestricted field of view for laser firing, only confined by the laser's range. This mechanism will be mounted to the top of the 2 meter by 2 meter by 2 meter cubic satellite. On the two opposing sides, foldable multi-junction solar panel arrays will be mounted to provide a constant source of energy to charge and power the spacecraft. Thirdly, the satellite will have a radar device to identify and track space debris from its location, allowing it to hone in on specific debris.
NASA's robotic servicing arm has a high range of motion with 7 degrees of freedom, allowing it to perform a wide range of tasks in space, including maintenance and payload handling. It also has mechanical joints that provide precise control and manipulation capabilities. Its precision makes it perfect to hold the laser. In this picture, the arm is shown extending from the space shuttle, known as the Canadarm, to position the Hubble Space Telescope during a servicing mission.
The laser itself would fire short, powerful 248 nanometer ultraviolet pulses at space debris. This causes ablation by vaporizing material from its surface, which creates thrust that slows the debris down. The reduction in velocity accelerates the debris, causing it to re-enter Earth's atmosphere and burn up quickly. The specific UV laser chosen for the satellite is a krypton fluoride laser. Through a pulsed power supply and a chamber of krypton and fluorine, the gases form an excited krypton fluoride molecule. As it decays to a stable, unexcited state, the molecules release energy in the form of a UV wavelength at 248 nanometers. This is represented by the equation shown at the bottom of the photo. Mirrors in an optical cavity increase the amount and intensity of UV light emitted. To reduce divergence or beam spread, a convex lens collimates the laser beam by bending light rays to make them parallel. This improves the precision of the laser, making it better at targeting space debris.
In order to ensure a space laser would work, calculations must be done to ensure its effectiveness. First, we must guarantee that the ablation threshold of aluminum is reached in order to actually impact the debris. The approximated fluorescence, around 100 joules per centimeter squared, is much higher than the minimum threshold of around 10 joules per centimeter squared of aluminum. Next, from the mass removal calculations and impulse calculations, we can approximate how much we can actually slow down the debris by per laser pulse. As you can see, the velocity of the space debris can be altered by roughly 0.4 millimeters per second for every pulse of laser light shot through the laser. This would equate to a change of 0.4 centimeters in its velocity every second, and this amount is significant to the point where the ablation technique by using space lasers is a viable remediation strategy.
In practice, the satellite will use its radar to locate space debris in low Earth orbit, which ranges from 200 kilometers to 20,000 kilometers. Once the radar completes the data collection, it fires the UV lasers towards the target. The satellite will turn off when it either completely removes space debris from LEO or becomes inoperative.
So we used Python's matplotlib and NumPy libraries to simulate a two-dimensional model of orbital debris. Each particle was given a randomly initialized orbital velocity and position orbiting the Earth. The blue sphere represents the satellite, and the red particles represent space debris. The laser was represented by a line connecting the satellite and the targeted debris, and the D-orbit was shown with the debris disappearing. Additionally, the algorithm draws the angle and distance required for the laser to hit the particle and ensures that when the distance between the debris and satellite is too high, the laser does not end up being fired.
Before deployment, there need to be existing agreements and cooperation between global organizations such as the Space Surveillance Network, European Space Agency, Federal Communications Commission, Federal Aviation Administration, and the National Oceanic and Atmospheric Administration. Additionally, funding would be required to launch the satellite, and we'd have to ensure the laser wouldn't target or accidentally fire at currently operating satellites. This would require safety protocols and real-time monitoring.
Once the satellite has been deployed, the continuing removal of space debris will be less expensive in comparison to the upfront launch. The life expectancy of targeted debris would be reduced, and once the debris entered the atmosphere, it would burn up completely due to its smaller size. There would be no risk of the debris impacting populated areas.
Ultimately, the Sigma satellite proposal would be the most cost-effective option for a remedial solution, It would reduce the amount of space debris between 1 and 10 centimeters in low Earth orbit by using ultraviolet light to ablate the debris. The project ensures that the debris is incinerated upon entry into the atmosphere.
In the future, extensive testing both on the ground and in space would be performed to validate the effectiveness of UV lasers in different operational conditions and refine the technology based on test results. This would be applied to make the excimer laser as energy efficient as possible. Additionally, different methods to target the laser would be studied. Aside from existing technologies such as a mirror, an internal gyroscopic mechanism would be evaluated where gears could be shifted in order to change the momentum and angle of the satellite. This would let the laser be affixed to the satellite and allow it to still be targeted when the full system rotates. As more space debris enters orbit, our proposal will continue to be modified and improved. Thank you guys for watching. We'd also like to thank Ms. Tomlin, Ms. Baggio, and Ms. Miller for their expertise, as well as the helpful mentors of Jimmy Barker and Mauro Esquivel. Additionally, Doug Frank from NASA was a great help, and Mariba Jah was a great inspiration.
My experience in the SEAS program were unexpected and significant. At first, I felt nervous about being grouped with complete strangers from around the United States. But as time passed, I was able to connect more with my teammates. Eventually, I considered them good friends, and them and I were able to complete our projects and learn more about space science.
My participation in the NASA SEAS summer internship was an amazing experience. From iFly to late nights researching, I had a blast throughout the entire week. I was fortunate enough to meet such an amazing group of people, including my teammates and mentors, who all made this experience such a wonderful one.
My engagement in the SEAS program will have a lasting impact on me, from exploring future paths in space science to making new friends. After touring the NASA Johnson Space Center and flying at iFly, I have been able to have amazing experiences with my teammates. Me and my team were able to work together and brainstorm our project, which deepened our friendship.
Being a part of the Space Debris Solution team on the SEES internship program has left a lasting positive impression on me. I met some really cool people, and my teammates have become family to me. Although the early mornings were quite rough, I truly enjoyed all the exciting places we visited, like iFly and the JSC. And working together to brainstorm experiments and work through problems was enjoyable with the help of my teammates. I'm truly grateful for my time at SEES.
Being a SEAS intern was impactful and a growing experience. At first, I was nervous and uptight, but as soon as I met my group, we all clicked, and as the days passed, we grew closer. This experience had me learn different ideas and perspectives in multiple genres, and I will be forever grateful for it.
As for me, I made some of my greatest friends at NASA SEAS, and I will be forever grateful to this experience for letting me make those friends. I also was able to connect with many NASA mentors and grow my worldview as well as my connections with others. Additionally, during the evenings when we weren't working on this very presentation, we also played pool, and it was really cool to make those connections between the research we were doing and the trajectory at which the cue ball had to be fired into the rest of the balls to make sure they went into the hole. I will never forget my time here, and I will cherish all the opportunities that come of this research. At this time, we are now open to questions. Please submit them through the Slido.
So we invite all members of the team to please open their cameras and unmute their mics. The first comment is, wow, what amazing research. Did you come into the internship with a knowledge about space debris?
I could answer that one, I guess. And I'd have to say no. We were only assigned this topic, I would say maybe a week before we actually arrived. And we really didn't have any knowledge about these specific techniques used to either mitigate the impacts of space debris or completely get rid of them in our Earth's orbit entirely.
So next question, were other ideas thought of on moving debris other than a laser, like sound waves or electromagnetic waves, or were they not as efficient, and why?
I can take this one. So yes, we explored various remediation techniques, including electromagnetic waves, a net, and a harpoon. However, through like a cost analysis and just an efficiency analysis, we found that overall a space-based laser would be the most cost-effective and efficient solution in remediating space debris.
This topic is globally important. Do you plan to continue this research with your team in the future?
I can answer that one. So currently we did a ZQube project testing how our debris would move in microgravity, and we will be getting that info after April. So we will be researching more on that topic and seeing how we can open it up to more resources.
Awesome. And how would you pitch this idea to possible investors or to NASA?
Right, so NASA's already done the research, and I think in our pitch we would definitely emphasize that although the initial cost is quite high, a laser in general is the most efficient at removing debris in space for each kilogram of debris removed. And at the same time, we're working ahead to prevent future costs of satellite repairs in the event of an impact or collision. And we're working to protect our astronauts on the ISS and potential future launches of spacecraft.
So we have a question for Ricardo. What was your favorite part of the research project?
Well, my favorite part of the research was being more with the team and experiencing like doing ideas off each other. Since we have like different backgrounds, we're able to each come up with different ideas, and at the end we decided on the space-based laser.
Well, you did an excellent job, so congratulations to your team. Our next team is the X-59 Lowering the Sonic Boom team, mentored by Rhonda McCarthy, a STEM educator from Des Moines, Iowa. She currently teaches computer science and STEM in rural Iowa at middle school. She loves to engage students in authentic space research and believes students are the key to solving problems for our future, both in space and here on Earth. Please welcome the Lowering the Sonic Boom team.
Hi, my name is Lynn Kohler, and I am pleased to introduce you to the X-59 Lowering the Supersonic Boom team. My team members consisted of Binyam Gebre, Aditya Kumar, Alejandro Zacharias, Anisha Malenti, Anj Doevedi, Ezekiel Peña, Isabella Noir, Mahalia Zavendra, Manav Patel. Our mentor who guided us on the project was Rhonda McCarthy.
As the idea of bringing back supersonic aircraft back into the industry becomes more and more prominent, the X-59 project came up as a partnership between NASA and Lockheed Martin to specifically reduce the loud sonic boom noise that is regularly experienced with the supersonic aircraft to a soft thump sound.
To be able to study exactly how the X-59 is able to reduce the sonic boom to a thump, a list of tasks and goals were outlined within this project. These goals consisted of analyzing the X-59's specific design elements to study exactly why each of the components were designed how they were and placed where they were. Also, by using mathematics and simulation testing to be able to understand aerodynamic flow, what kinds of forces are acting upon the aircraft, like lift and drag, and finding the areas of low and high pressure. Additionally, by constructing an Arduino board and supplementing it with various components and code to quantify the sonic boom produced by the X-59.
A comprehensive methodology was designed to effectively dilute and present the following information. Our team was divided into various tasks. The first section details our team's in-depth research into the X-59's exceptional engineering. The design enables extreme noise reduction capabilities. The second section presents detailed calculations based on extensive open source data showcasing the forces acting upon the aircraft. The model testing segment covers our development of an accurate 3D model and testing the model for airflow simulations to test general airflow. Finally, the data collection and analysis section again uses open source data to demonstrate the X-59's ability to produce a subtle thump when breaking the sound barrier.
Ever since the Federal Aviation Administration prohibited supersonic flight in 1973, there have been several attempts to minimize the effects of sonic booms, primarily because of the benefits and of reduced travel time. So this design on screen has an elongated nose which spreads out the shock waves from the aircraft. So first the air will hit the top of the will hit the tip of the nose and it will propagate aerodynamically towards the tail of the aircraft. Also, because of the irregular elongated nose, a camera had to be placed on top of the aircraft to allow the pilot to be able to see the outside. The engine was also positioned at the top of the aircraft so that most of the shock waves would be deflected towards the sky rather than the ground. Additionally, the inlet manages the airflow into the engine to prevent low energy boundary layer from being ingested and ensuring smooth airflow despite the top-mounted engine position. And finally, the aircraft incorporates a swept-back wing to be able to reduce drag. All of this makes the X-59 aircraft able to reduce the sound of a sonic boom to a thump.
In order to test the airflow of the X-59, we decided to try using a wind tunnel simulator. Initially, we started by downloading the X-59 model from NASA's website. However, it came with a stand that would skew the results. So, we imported it into CAD and 3D modeling software to remove it. We started with Onshape. However, the model had some faulty surfaces. We attempted to repair the faulty surfaces in Fusion 360 and Onshape. We managed to solve them in Fusion 360. However, the 3D model wouldn't work in our wind simulator, OpenVSP. So, we tried to convert the model into a usable model for OpenVSP. When OpenVSP didn't work, we switched to Blender.
When we switched to Blender, we started by removing the stand vertices. This gave us a successful STL that we would be able to use to simulate a wind tunnel. We tried finding a program that would help us create the wind tunnel, and we went through loads. We went through OpenVSP, OpenFlow, and Flow3D, but in the end, we ended up deciding to use Blender because all of the other softwares either would have taken too long to set up or they would have used too much processing power than we have available. So when we went into Blender, we attempted to simulate a wind tunnel, but when we did that, the resolution was too high and it was trying to simulate realistic lighting and the visual smoke was not working. So we redid the process in Blender and we got a wind tunnel working at a resolution where you can tell what the smoke is doing pretty accurately and there's no realistic
Due
light.
to the limited results from the 3D simulation software used pertaining to lift and drag data, the team used the modern lift and drag equation to simulate the possible lift measurements based on standard data values. As NASA doesn't release a specific coefficient of lift of the X-59, the team used a similar aircraft called the C-25D, which includes the same characteristics as the X-59, such as the long nose and engine placement being at the top and the swept back wing. Additionally, the team used the ICAO standard measurements for air density at 55,000 feet to use as an accurate measurement, as there isn't currently data released by NASA regarding air density of where the X-59 would be flying in. After performing the calculations based on the X-59's velocity and cruising altitude, the team formulated the lift of the X-59 to be 13,725 pounds of force and the drag force being 8,286 pounds of force. To be able to assess which phase of flight the X-59 could be in, the team referenced the coefficient of lift versus alpha graph. The corresponding angles of attack as a result of the low coefficient of lift measurements would be a negative angle of attack. Since, based on the data, the weight of the X-59 is greater than the force of lift, also the force of thrust being greater than the force of drag, and the angle of attack being negative, the team hypothesized that the X-59 could be inside
The images to the right show the pressure distribution at different sections of the X-59 in supersonic flight. The first image displays a colored palette of pressure when the X-59 travels at supersonic speeds. These colors are valued to show higher pressure at red and lower pressure at blue. As you can see, the cone shape is formed as the aircraft is moving at supersonic speeds and is moving faster than the sound waves. Additionally, you can see higher amounts of pressure measured in pascals at the nose of the aircraft and the rear where the boom dissipates. The data shows the high pressure shock waves at the nose followed by low expansion waves at some places, such as the inlet located at the rear of the plane, which experiences spillage resulting in higher pressure in the area indicated. The second graph shows a monochromatic distribution of the surface pressure in a simulate when the X-59 travels at supersonic speeds. Just as before, the cone shape is formed as the aircraft is moving at Mach 1+. The differences in pressure can be explained based on the position in which shockwave and expansion
We
occur.
needed to devise a testing plan utilizing the Arduinos for microphone experimentation. Several people in our team worked on coding the Arduinos and Cduino, and we successfully lit an LED light that reacts to sound waves. We then calibrated different frequencies of instruments to ensure the microphones functioned correctly. After the calibrations were successful, we simulated flight data by surrounding a plastic box with bubble wrap. We inserted different microphones into the box and played the sonic boom on a Bluetooth speaker to collect the data for analysis.
During the initial phase of our Arduino project, we connected an Arduino Uno to a breadboard and implemented LED lights to confirm the functionality of our microphone. We tested various microphones, both built-in and external, and explored their respective qualities. Next, we added an SD card reader to store data on an SD card. We collected specific data on the sound, amplitude, and frequency of each microphone. Our observations revealed that some microphones were more sensitive than others. This detailed data allowed us to understand the strengths and limitations of each microphone. The more sensitive microphones showed promise for applications requiring the detection of subtle audios, while others might be better suited for environments with higher noise levels. Additionally, we experimented with various configurations and placements of the microphones to optimize their performance. For instance, we tested different angles and distances from the sound source to determine the optimal setup for maximum sensitivity and accuracy. This iterative process of testing and refining our setup ensured that we achieved the best possible results for our project.
We had a couple people working on already assembled microphones while I tackled building one completely from scratch. I used a microphone with 2 strings and worked with attaching all the components. For future scientists, I would not recommend this microphone as it was very involved and there were some code malfunctions with it.
This code is designed to use the Arduino to detect sound and indicate the detection through an LED. 2 key components are connected to the Arduino, a sound sensor on digital pin 2 and an LED on digital pin 3. At the top of the code, we have these 2 components declared and initialized. In the setup function, the pin mode function configures the sound sensor pin as an input, meaning it will read data from the sensor, and the LED pin as an output, meaning it will control the LED. The loop functions runs continuously, constantly checking the status of the sound sensor using the digital read function. If the sound sensor detects a sound indicated by a high signal or 1, the code turns on the LED by setting the LED pin to high using the digital write function. If no sound is detected, indicated by a low signal or 0, the code turns off the LED by setting the LED pin to low. Here's a demo of this code.
This is the code to make the SD card reader function. The first arrow is pointing to the code that initializes the SD card, gets it started, and able to be written to. The second arrow is pointing to the code that creates and opens the file, as well as adds the titles of the 2 columns that will be frequency and amplitude. The third arrow is pointing to the code that converts the data onto the CSV file, and the fourth arrow is pointing to the code that uses the digital write command to turn the LED on and
In
off.
Arduino Phase 2, we worked on a sealed container with bubble wrap insulation. We used a sealed container so no silent waves would escape. We also insulated the container with bubble wrap so no echoes would affect our data. In the box, we had our speaker and Arduinos that would emit and pick up the sounds. With our variables, we had independent variables, the microphones, and frequency and amplitude provided. Our dependent variables was frequency, amplitude, return. Our controls were sealed container, bubble wrap insulation, Arduino board, and the distance between the speaker and the microphone, which came out to 52.5 centimeters. Regarding the process, first we made sure the speaker worked, so we tested frequencies from 200 Hz down to 100 Hz. From there, we were able to get a consistent data, so we knew the speaker worked. From there, we tested 20 and 40 Hz on the microphones.
When performing the calibration readings at 20 Hz and 40 Hz, there hadn't been consistent and valid results with the microphone. These issues could be attributed to a multitude of different reasons, such as having a poor speaker that couldn't have emitted a great sound, or a frequency checker that wasn't able to quantify the low frequency sounds accurately, or not getting the correct sampling rate for the Arduino board. So, the team simulated what the frequency versus amplitude graph would look like to the right. The team would expect there to be peaks in amplitude at 20 Hz and 40 Hz, as those were the frequency sounds that was used to calibrate the instrument. The reason for picking to calibrate the instrument at 20 Hz and 40 Hz is because most of the energy that is produced in sonic booms are concentrated at a frequency range of 0.1 Hz to 100 Hz.
From the Arduino testing, we wanted to get the pressure versus time and amplitude versus frequency graphs for the X59. The human ear can hear at a frequency range of 20 to 20,000 Hz, with it being the most sensitive to frequencies in the range of 1,000 to 3,000 Hz. Anything below around 50 Hz is very difficult for the ear to detect. Dr. Kevin Crosby provided us with 10 simulated flight overpasses of the X59, and they are simulated with the standard IKO atmospheric model with the X59 in the cruising stage. The amplitude versus frequency graphs for the 10 simulated flight overpasses shows that most of the sound produced at frequencies above 50 Hz, which are very easy for the ear to detect, have very low amplitude, and are thus imperceptible. The amplitude is high for frequencies below 50 Hz, which are very hard to detect. What all this means is that the X59 has a very low sound profile for the human ear. The pressure versus time graph for the 10 simulated flight overpasses also shows that the end pressure wave graph has more rounded peaks than those for other supersonic aircraft, which shows that the sonic boom is softened and the waves are more spread out to reduce extreme pressure.
We compared the prices of the microphones tested on the different Arduino and Arduinos to their data collected. This red high sensitivity microphone is attached to the Arduino, and it is the most expensive of the microphones tested. This expensive microphone has a tendency to have a great digital but almost useless analog, and its data proved this by not providing significantly finer data than any of the other microphones tested. But it also required a lot of sound to light the LED, so we decided this microphone is not worth the price. We also tested the cylindrical electret condenser microphone on an Arduino. It's simple, reliable, and inexpensive microphone with just 2 wires. We were unfortunately not successful in the timing and coding of this microphone. We have tested several codes, but we could not just make it work. The best microphone we tested on the Arduinos was the blue high sensitivity sound microphone, which is deemed to be really reliable. It picked up our voices as we were speaking in the room, meaning it was extremely sensitive, making it a fantastic microphone to measure the sonic boom. It isn't as expensive as the red high sensitivity microphone, and for its data, we concluded the blue high sensitivity sound microphone would be worth the price.
Great job. So we invite everyone from the X59 team to turn on their cameras and unmute their microphones. Our first question is, how was the LED light helpful with your Arduino testing?
I can take this one. So we use the LED light as a way of testing to make sure our microphones are working. So if the microphone did pick up on sound, then the LED pin would be set to high, and that's when it would turn on.
Great. Did you know how to use CAD or 3D modeling prior to coming to the SEAS program?
Okay, so I got this one. I'd used Blender a little bit, and other people had used other softwares in the past, but we really had to learn a lot of the softwares by scratch from the documentation during the process.
Would you require new specialized training for pilots flying the X-59 aircraft?
So currently there is, I'm pretty sure only 1 or 2 pilots that are capable of flying the X-59. And so in the future, to do more testing, it would probably make it easier if a couple more pilots were trained, because that would mean that the X-59 would be able to go and fly more often.
Great. And if someone wanted to invest in your project, what would your pitch be to investors?
I could answer that question. So the X59 is able to achieve supersonic speeds at while simultaneously generating low amounts of noise and low amounts of sonic boom noise. So and also it's very cost effective. As we mentioned earlier in the presentation, as our microphones was very low cost. But yeah, that would be mostly our pitch because it's able to produce such low amounts of noise in comparison to other aircraft that goes at similar velocities.
Excellent. Your team did a great job. Congratulations. The success of SEAS is determined not only by the commitment and research conducted by our student interns, but by our mentors, project assistants, and project associates. To our NASA subject matter experts, mentors, project assistants, your passion for youth development and STEM education and learning and NASA missions and opportunities is to be applauded. I know the SEAS interns join us in thanking you for your dedication, guidance, and commitment. A special thank you to the CSR administrative staff for their assistance during the SEAS internship program. A special thank you to Zoe Zlatic, a SEAS alumni and freshman at the University of Southern California, where she studies astronautical engineering. Zoe was selected to fly her SEAS microgravity research on board the zero-G parabolic aircraft. She is a peer mentor to this year's SEAS interns and is an aspiring engineer and astronaut. This concludes the NASA STEM Enhancement in Earth Science Symposium for our rising junior student interns. Thank you for watching these amazing students as they share their results of their internship and their research. The future certainly looks bright for these amazing students. So congratulations to all of you, and we cannot